receptor family, and it regulates outgrowth and stabilization of peripheral microtubules. It's an enzyme, so most of the functional proteins have an enzymatic activity, but in addition to that, they have some other functions. For instance, in this case, this protein acts as a transcription factor, and it is involved in transcription of ribosomal RNA genes by RNA polymerase 1. And these are the references that show what's the role of these proteins in different cells. There is another example, and that is glucose 6-phosphate isomerase, or abbreviated as GPI. And this enzyme, probably you are familiar with this enzyme because it's an important enzyme in glycolysis. When it is present as a dimer, it catalyzes the reversible conversion of fructose 6-phosphate to glucose 6-phosphate. But when it is a monomer, it acts as a neurotrophic factor, and it promotes the survival of embryonic spinal neurons, skeletal motor neurons, and sensory neurons. So let's talk about now alcohol, how alcohol affects our body. So it really depends upon how we consume alcohol, acute versus chronic. So if we consume rapidly excessive amount of alcoholic drink or beverage, that is referred as acute alcohol consumption. And this rapid consumption of alcoholic beverage depresses the central nervous system function, leading to slurred speech, meaning that you won't be able to understand what the person is saying who has consumed alcohol. Or there is an incoordination of muscles, so the person cannot walk, or they will feel drowsy, or they sometimes they can lose consciousness. On the other hand, chronic alcohol consumption is due to excessive consumption of alcohol. It can be either due to heavy drinking, which indicates that, which means that 15 drinks or more per week in men, or 8 drinks or more per week in the case of women, as opposed to the binge drinking, or sometimes it is called binging. This is mostly seen in college students who, at the end of the Friday evening, they are all in the pub sitting and drinking, and so they will drink over the weekend, and then Monday come, they are not fully aware of what's going on. So every week they will follow this pattern of consuming large amount of alcohol. So this is called binging. So if we drink moderately, meaning that if we occasionally have a glass of red wine, it is considered to have beneficial cardiovascular effects. But if we drink over a long period of time, or we do binging, it has serious health issues, and it can lead to chronic diseases, including death. So unhealthy alcohol drinking pattern has effects on brain, and it eventually leads to compulsive alcohol use or dependency on alcohol. Earlier days, it used to be referred as alcoholism or alcohol addiction, but now it is referred as alcohol use disorder. And how this occurs? It is, I think it's the following slide. So it's the chronic consumption of alcohol not only affects brain, but it also affects several other organs and organ systems, as shown in this slide. Apart from having an effect on different organ system, there are certain behavioral changes in AUD, or alcohol use disorder. And women are more susceptible to alcohol's effect than men. This is because of the differences in absorption of alcohol, or how our bodies metabolize alcohol. So what are the different effects of behavioral changes, or changes in the behavior of people? It could be confusion, slurred speech, impaired judgment, motor impairment, impaired memory, unstable moods, or sometimes it can be violent behavior or inappropriate behaviors, which can include sexual behaviors. And often when they consume large amount of alcohol, it can lead to blackouts, or they can experience blackouts. In blackouts, when person experience blackouts, they do not remember the events or what happened, where they were, what they were doing. If the blood alcohol is very high, it can lead to coma or even death. The person can die because of excessive alcohol consumption. So what are the different levels of alcohol that have these effects? So if we have 0.02% alcohol in our blood, then we feel a little warmer and we begin to make poor judgment. But if we have alcohol equal to 0.05%, then our ability to focus is not there, is no longer there, and we begin to have a blurry vision. And at 0.1% alcohol, or blood alcohol level, abbreviated as BAC here, speech will be slurred. And at 0.3 or 0.3 to 0.4%, people can experience blackouts. 0.2 to 0.29, they experience blackout, and 0.3 to almost 4, they can be unconscious, and it can potentially lead to their death. But if the blood alcohol levels are 0.4 or bigger than this, then it can lead to coma or sudden death. So in US, the legal limit is 0.08. If the concentration is above that, you cannot, it is considered unsafe or illegal to drive. In India, the concentration is 0.03%, which is far less than what we have here in the US. So you really need very little alcohol in your system to experience these side effects of alcohol. So what happens? So these are the effects that you saw due to alcohol present in the blood. But what happens when there is a withdrawal, when the blood alcohol level decline in our system? So this can be experienced within few hours to few days, meaning that the alcohol withdrawal can be experienced within few hours of stopping alcohol consumption to few days. Such, during alcohol withdrawal, individual experiences sweating, rapid heartbeat, tremors. Tremors, probably you have seen, handshakes. Hand is shaking as if there is some nervous issue, but it's probably because they lack their body needs more alcohol to calm their nerves down. Or they have a problem falling asleep, so they will say, okay, I have to have another drink to fall asleep. Or they can be agitated, nausea, vomiting, hallucination, etc. So these are all the effects that are seen in these individuals. So what they do, then they continue to drink more alcohol. And the more severe these symptoms become, their ability to function properly at work or in social situations is impaired. So, they will behave improperly at a social gathering, for example. And so how these effects are achieved. So most of the things that I have shown in the previous slide due to withdrawal of alcohol in the system are more or less regulated by what happens in the brain. In the brain, there are several neurotransmitters, and they are responsible for proper functioning of neurons and non-neuronal cells in the brain. So I'm going to mainly focus on glutamate. Glutamate is a neurotransmitter that binds to glutamate receptors. So what happens in alcohol use disorder to glutamate receptors? So normally in a naive individual who has not been exposed to alcohol, there is always a balance between GABAergic inhibition and glutamate excitation. So you can imagine glutamate receptors are excitatory and GABA receptors are inhibitory. So there is always a balance between the two. But once the person or individual begins to drink alcohol, this balance tips towards glutamate. So glutamate goes down, and GABA inhibition is stronger. And such individuals feel anxiolytic effects, and they feel sleepy or sedation. There's sedative effect. However, once the individuals begin to drink more alcohol, then the balance is tipped to the other side. Now the GABA inhibition is stronger, or it's less stronger, but glutamate excitation is much higher. And so these effects will be seen when alcohol is not in their system. So they will experience anxiety, hyperactivity, not able to fall asleep, insomnia, or their hands will shake, and that is tremors. So to calm their nerves, they will begin to drink alcohol. So this is on a daily basis. Once the morning their alcohol is out of their system, by afternoon or evening, late evening, they will begin to drink alcohol again. So they will fall asleep. Next day, the cycle begins, and this process leads to birth of an alcoholic. And alcohol affects rich and poor alike. And I brought one good example that you probably already know, and that's film actress Meena Kumari, my namesake. She died because of alcohol use disorder. So now I'm going to take you to glutamate receptors because that's where my research has been focused. So glutamate receptors are broadly classified into ionotropic and metabotropic. Ionotropic are ion coupled, and they are further classified into NMDA receptors, AMPA receptors, and kainate receptors because they bind to ligand NMDA, AMPA, or kainate. So my talk is mostly focused on NMDA receptors. NMDA receptors consist of GLU-N1. So previously it was called NR1. This is a product of a single gene, and it has 8 splice variants. The second subunit is a family of 4 subunits. It's called GLU-N2, and it has 4 subtypes: GLU-N2A, N2B, N2C, N2D. And then we have a third subunit that is GLU-N3. These are mostly seen during embryonic development. And these are further 2 types, GLU-N3A and N3B. In alcohol addiction, or AUD, GLU-N1 and GLU-N2B are the ones that are upregulated. Sorry, there may be some slight shift because I had to upload my PowerPoint into Google Drive. So there may be some shift in these highlighters that I want to point out here. The NMDA receptors, let me just go back a little bit. The GLU-N1 is the obligatory subunit. It has to be present in order to make functional NMDA receptors. So GLU-N1 will combine with members of GLU-N2 or GLU-N3 to form functional receptors, which is shown in the next slide. Here, this is a cartoon. This is the plasma membrane, which is a double bilipid layer. And here are 2 subunits of NR1, one subunit of NR2B or NR2A, or GLU-N2A or GLU-N2B, and GLU-N1. So these are tetrameric. Some people believe that they are pentameric, but mostly people believe that they heteromeric and functional NMDA receptors are tetrameric in nature. So this is the cytoplasm, and this will be the cell surface. So NMDA receptors, why they are important? They mediate, as I mentioned earlier, they mediate excitatory neurotransmission in the brain. They are very important during neuronal development, synaptic plasticity, and long-term potentiation. So NMDA receptors consisting of GLU-N1 and GLU-N2B are the first ones to appear. In the case of mice, they appear during embryonic stage 11.5 days. So when the embryos are 11.5 days old, then and neurons are just differentiating, these 2 subunits are the first ones to appear. The NMDA receptors are voltage dependent. They are highly permeable to calcium, and when These receptors are overstimulated. It leads to neurotoxic effects, which eventually leads to cell death. And probably this explains why the brains are so small in individuals suffering from AUD. Chronic ethanol, as I mentioned, chronic ethanol exposure upregulates NMDA receptors, especially consisting of GLU-N1 and GLU-N2B subunits. And this effects had been confirmed many times by so many different labs using both in vivo models and in vitro models. And it is believed this upregulation contributes to the development of alcohol dependence and withdrawal syndrome. So let us see how chronic alcohol consumption upregulates these NMDA receptors. So I will talk of GLU-N1 and GLU-N2B because these are the ones that are affected by AUD or chronic alcohol exposure. So if considering what is present in the control neurons is 100% protein. So whatever the levels of GLU-N1 subunit is, that's 100%. So when we compare neurons following chronic ethanol exposure, there's a significant increase of GLU-N1 and significant increase of GLU-N2B. So there is a high expression of these receptors. But when we look at the RNA, there is a significant increase of GLU-N2B mRNA, but just a nominal increase for GLU-N1 subunit. So my goal was to see what is the mechanism that have this differential effect of alcohol on neurons. So we used rather an in vitro model system of fetal cortical neurons where these neurons are cultured and exposed to 50 millimolar ethanol for 5 days, and then the experiments were conducted. And whenever it was possible, we used an in vivo model system of C57 black 6 mice. They were given 7% alcohol for 10 days and a small amount of chow, which prevented bleeding in their stomachs. And here is a C57 mouse drinking alcohol. So the first thing that we conducted experiments to see whether something in the nucleus is happening and something happening in the cytoplasm. So we looked at the mechanism in the nucleus and in the cytoplasm. And I'm just going to give you the results of this. So we concluded from our study that chronic ethanol exposure of neurons, cortical neurons, increases the half-life of GLU-N1 mRNA, and at the same time increases GLU-N2B gene transcription rate. So in one case, it is the stability of mRNA. In the other case, it is the rate of gene transcription. So now I will mainly talk about GLU-N1 mRNA. So my goal was to see what is causing to increase the half-life of GLU-N1 mRNA. So the first experiment we did was look at the effect of cyclohexamide. Cyclohexamide, as you know, inhibits new protein synthesis. So when we determined the half-life of GLU-N1 mRNA in the presence of alcohol and cyclohexamide, we were able to calculate the half-life of GLU-N1 mRNA, which was 22 hours, as opposed to chronic alcohol in the absence of cyclohexamide, where we could not see the levels coming down. So that means that new protein synthesis is somehow involved in stability of GLU-N1 mRNA in response to chronic alcohol consumption. So normally, the regulatory units that are involved in stability of mRNA are located in the 3' untranslated region. And whereas the 5' untranslated region, which is shown here in this black little line, is involved with translation of the mRNA. So if there are certain cis-acting regions present within the 3' UTR or 3' untranslated region that binds to transacting factors. So we thought that this could be proteins because new protein synthesis inhibits stability of mRNA. So we set out to find out if truly there exist cis-acting regions in the 3' UTR of GluN1 mRNA, and if there are specific transacting factors or proteins that bind to such regions. So the first experiment we conducted was we amplified the 3' UTR of GluN1 mRNA. Sorry, this should be read GluN1. And then we isolated polysomes and polysomes from control and ethanol exposed neurons. And then they were combined, incubated, and separated on a gel. And this is RNA mobility gel shift assay. So as you can see, when we have the proteins from control and ethanol exposed neurons, the probe, which is the full-length GluN1 UTR, is migrating slowly in these two lanes. C is the control neurons and E is the ethanol-exposed neuron. And the free probe is right here. Because it is such a large, it's around 1,000 nucleotides, so I had to use agarose gel for this assay. So now we know that there are cis-acting regions in 3' UTR and also transacting proteins binding to this region. So the next question was, what is the location of these cis-acting regions? So we chopped this full-length GluN1 3' UTR into several fragments, and we tested them using the same RNA mobility gel shift assays. With the exception of delta 5, so this is delta 1, delta 2, delta 3, delta 4, and this is delta 5. So delta 5 is right here. There is no shift in this, but with all the other fragments, we saw a clear shift. What we observe here, that it is a smear, it is not discrete bands. So whether it is now one protein or multiple proteins, that was my next question. So before I could do that, I had to first resolve this smear. So we used two alternate techniques. One was UV crosslink analysis, and I think I have to go back a little bit. So since this region, the delta 4 region, was common to all the fragments, so we thought the cis-acting region is located in this particular region. It's around 156 nucleotide long. But subsequently, we found out that there are 3 additional cis-acting regions, 1 in this and 2 in this region. But today I'm going to show only results that were obtained using delta 4 region of GluN1 mRNA. So the first technique that we used is the UV crosslink analysis. And we used, of course, delta 4 RNA for this. And we found 3 bands that were highlighted with this technique. And they had this molecular weight. Initially, we thought that this lower radioactive band is a free probe, but subsequently, I think it took us 3-4 years to figure out that this was another protein that were binding to delta 4, and it's around 10 kilodalton. And then we confirmed these results using another technique. Slide stopped moving somehow. Just bear with me. It's for some reason it's slowed down, and it's
no
not.
Issue. Yes, now it is moving. You can proceed now.
But it doesn't show me here.
Over the screen, it is moving. We can see next slide.
Showing UV crosslink analysis.
All right.
Okay. So, so then the next technique, oh, it moved too much now. Okay, so then we use the second technique, which is Northwestern analysis, and we use again delta 4 RNA. So when we exposed the membrane, we highlighted only one band after overnight exposure. But when we exposed for a longer time, then we detected two additional bands in this area. And we could compete all these bands using non-labeled or non-radioactive delta 4 RNA. And this band was clearly, present in large excessive amounts in ethanol exposed neurons. So we quantitated this, and it was significantly higher intensity of 88 kilodalton band. So of course, this is an ethanol sensitive protein, RNA binding protein binding to GLUN1. So we wanted to know its identity. So we use this protocol to purify the protein, and then we perform the mass spec analysis on the purified protein. And there were 6 peptides that matched to beta subunit of glucosidase 2, abbreviated as G2 beta. So this is an enzyme, part of an enzyme. So we were not sure if it is correct. So we got the antibody from our collaborators, and we performed RNA mobility shift supershift assay. In this, it's the same kind of setup as RNA gel shift assays, except we add increasing concentration of G2 beta antibody. As a control, we use NRS, normal rabbit serum, because G2 beta antibody was raised in a rabbit. So with the increasing concentration of G2 beta antibody, we saw a very nice supershift with a concomitant decrease in the intensity of the RNA protein complex, confirming that it is indeed present in the RNA protein complex. Then we performed another technique where we immunoprecipitated G2 beta protein and processed it for RNA isolation. And isolated RNA was used as a template for RT-PCR, reverse transcriptase polymerase chain reaction, using GLUN1 gene specific primers that span exon 20 and exon 21. And in this case, we use the mRNA transcribed in vitro from GLUN1 as a positive control. So with the positive control, we get one band, but in the immunoprecipitated RNA, we got 2 bands, which are GLUN1 and GLUN1 These are the 2 full-length splice variants that are expressed in cortical neurons. So this indicates that truly, yes, G2 beta is a RNA binding protein, and it binds specifically to GLUN1 mRNA. So here is a cartoon that shows, in the absence of, in control neurons, G2 beta binds to delta 4 cis-acting regions. But once you give the alcohol, then the amount of G2 beta is increased in this location, preventing exonucleases to attack GLUN1 mRNA for its degradation. And then few years later, after identification of G2 beta as GLUN1 mRNA binding protein, we purified the second protein, which was 47 kilodalton, and that turned out to be annexin A2. So this is the second GLUN1 mRNA binding protein. And this is also a multifunctional protein. It was, when we identified this as a RNA binding protein, it was already known that it binds to its own mRNA, and it also binds to c-MYC RNA, But in both cases, it binds as a tetramer, but to glue and one mRNA, it binds as a monomer. And annexin A2 is involved in exocytosis, endocytosis, membrane organization, ion channel conductance. It links effectin to cytoskeleton to the plasma membrane. So you can see that annexin A2 is also a multifunctional protein. I'm not going to talk about annexin A2 subsequently. So that's why I'm mentioning it here, that it is also a multifunctional protein. It's an RNA binding protein, and apart from this, it's involved with many cellular processes, and especially the effectin, its association with effectin cytoskeleton is involved with translocation and transport within the cells. So just briefly about G2 beta protein. It was first identified as a substrate for protein kinase C. So because it was not known what it is, so it was abbreviated as PRKOSH, because it is a protein kinase C substrate. And it's an acidic phosphoprotein because it's obviously it is heavily phosphorylated, so it will be acidic. And it anchors the catalytic, this is much later. The first identification was in 80s, and in 96 it was identified that it binds to, or it anchors the catalytic alpha subunit of alpha glucosidase 2 in the rough endoplasmic reticulum. So which means that G2 beta is responsible for retaining G2 alpha subunit in the rough endoplasmic reticulum. And the alpha glucosidase 2. So alpha glucosidase 2 consists of G2 beta and G2 alpha. Both together form an enzyme called alpha glucosidase 2, which is an important enzyme in protein folding. And we will talk more about this. And briefly, the structure of the beta sub G2 beta. It has a leader sequence. This green end on this side is the N terminal, and on the 507, where it is indicated 507, that's the C terminal. At the C terminal, it has HDEL sequence that helps it associate itself with the endoplasmic reticulum membrane. So that's how it gets associated with the ER membrane, and then it binds to G2 alpha using these two domains, this blue one and the light blue one. The dark blue and the light blue are the two domains that interact with G2 alpha. It also has EF hand that binds to calcium, and box B1 is the region that is alternately spliced, but in neurons, G2 beta is not alternately spliced. The green one is the MRH domain, which is very important. It assists glucosidase 2 enzyme in hydrolyzing glucose. And these two arrows indicate where putative and linked glycosylation sites are. And this purple box here in the center is the acidic region. There are many glutamic acids. It's a glutamic acid stretch. So it gives negative charge to the protein. And that's why the G2 beta, even though it is 54 kilodalton protein, but it migrates as 84 to 90 kilodalton, depending upon which manuscript you are looking at. It migrates as 80, 82, 84 to 90 kilodalton protein. So if it is, if it is facing the ER lumen, so this is the cytoplasm. What you are looking at here is the ER membrane, endoplasmic reticulum membrane. So this is the cytoplasm site, and this is the lumen of the ER. So HDAL helps G2 beta gets associated with the ER membrane. Then it binds to the yellow, these yellow dots indicate this G2 alpha. So it's an important enzyme in protein folding. But if it is binding to RNA, it has to be outside the ER, facing the cytoplasm. So this will be the lumen of the ER, this will be the cytoplasm. The reason being, mRNA never enters the ER lumen. It's always outside. So when the newly proteins are synthesized, they get translocated into the ER lumen. And we'll talk more about this. So we learned earlier that it is an ethanol sensitive protein. So we wanted to check how chronic ethanol affects G2 beta polypeptide levels. The previous technique really was due to binding of delta 4 RNA. But now we wanted to check on the Western blot whether it truly is upregulated or not. So when we examined by Western blotting, surely the quantitation of these Western blot indicated that it is significantly upregulated in ethanol exposed neurons. So the question is now, what's the fate of this increased levels of G2 beta in neurons? Could it translocate to nucleus? The reason we ask this question to ourselves is because there was one publication where from using breast cancer cells where they found G2 beta translocates to nucleus. So there it could bind to RNA and transport it to outside the nucleus. So that could happen, but we wanted to confirm that whether this is possible in the case of neurons, or it's a possibility. The second is that it could retain higher levels of G2 alpha, forming higher levels of G2 alpha, alpha glucosidase 2 enzyme, and that will increase the efficiency of protein folding. Or the third alternate was that it could interact with GluN1 mRNA and retains higher levels of GluN1 mRNA in the ER. But we observed that.
May I interrupt, please? I'm sorry to interrupt, but ma'am, the audio is very soft. If possible, if you could please be a little louder, it'd be nice. Ma'am. Sorry to
Okay,
interrupt
I
again.
increased the volume a little bit. Can you hear me now better?
Ma'am, I can hear you already, but I think the audio is a little, it's too soft, so we can't hear you very properly.
Can you hear me now?
Yes, ma'am, it's much better now.
Okay.
Please go ahead. Thank you.
Okay. So there are three. We wanted to see what happens to increase levels of G2 beta in neurons. So there are three possibilities. One, it could translocate to nucleus. Being RNA binding protein, it could help bring the newly synthesized mRNA to the cytoplasm. And the second reason for this thought was that that in breast cancer cells, it is G2 beta translocates to nucleus. They did not see they did not do the follow-up study to see what G2 beta is doing in the nucleus, but surely there was some indication that it could translocate to nucleus. So that was first thought that came to our mind. Second was that whether it could retain higher levels of G2 alpha subunit. This is one of the ligands of G2 beta. So if it retains more G2 alpha, it could form much more alpha glucosidase 2 enzyme. And then if that is the case, then the protein folding will be enhanced. It will be facilitated because that's one of the function of alpha glucosidase 2. As a third option, we thought that G2 beta could interact with GluN1 mRNA and retain higher levels of GluN1 mRNA in the ER. But we saw in one of the previous very early slides that the level of GluN1 mRNA did not increase significantly. It was just a nominal increase. It was 111% as compared to 100% in control cells. So that means that this option is not there, but we did test it out. So we tested these different possibilities. So when we looked at we purified the nuclei from control and from ethanol exposed neurons, and we performed Western blot analysis, we did not detect G2 beta protein in nuclear preparation. And as a positive control, we used nu N It's a protein that is that resides in the neuronal nuclei. So this option is out, that it does not translocate to nuclei in neurons. So then we looked at the G2 beta levels in S100 and P100. P100 is the polysomal fraction, which contains all the membrane fractions, endoplasmic reticulum, Golgi complex, endosomes, polysomes, lysosomes. They are all present in this fraction. And S100 is the pure cytosol, organelle-free cytosol. So we examined G2 beta levels, and we detected two bands. One was around 130 kD band, and the other one was the around 80 kilodalton or 83 kilodalton protein, which is the G2 beta protein. It turned out that the upper band is a dimer of G2 beta, and this is mainly present in the cytosol, not in the polysomes. And when we quantitated these levels, we saw a significant increase in ethanol-exposed lysate and in polysomes, whereas in the case of S100, there was a significant decrease in G2 beta protein. It's not very clear. Something happened when I uploaded my slides to a Google document. And then we examined the alpha subunit of glucosidase 2. And we used the same set of samples, and we did the Western blotting and analyzed and quantitated the levels of G2 alpha protein. There was a decrease in ethanol-exposed polysomes, but a significant increase in cytosol, which was very surprising to us. So the next question was whether G2 alpha, that is exiting or egressing the endoplasmic reticulum, is it catalytically active? So when we tested, we performed the alpha glucosidase 2 enzyme activity, and sure enough, in S100, the alpha glucosidase 2 is catalytically active, and there is a decline in P100, the catalytic activity of alpha glucosidase 2, with a concomitant increase in cytosol. So to summarize these results, G2 beta does not translocate to neuronal nuclei irrespective of the ethanol treatment. Ethanol exposure translocates G2 alpha from RER to the cytosol, where it is enzymatically active. And the third one, I did not share these data with you. When we incubated the delta 4 RNA with the polysomes, we could not displace G2 alpha from G2 beta, G2 alpha complex. But what this means, what's the impact of this information? So I have to take you to how protein is synthesized in the cells. So the nucleus is in this region, and here is the nuclear envelope. And as you know, endoplasmic reticulum is continuous with the nuclear envelope. So here is the endoplasmic reticulum, Golgi complex, and proteins for export are transported as vesicles. They either get transported out of the cell or they become part of the plasma membrane. If they are they are not properly folded, they can lead to lysosomes for their degradation. So now, if we just focus in this area in the next slide, it takes us to how the glycoproteins are synthesized in rough endoplasmic reticulum, which is abbreviated as RER. So the messenger RNA, which is present, what you are looking at here, this two lines, blue lines, indicate the endoplasmic reticulum membrane. And these are the blue circles or blue cylinders are the translocon that are formed of Sec61. These help transport the proteins inside the endoplasmic reticulum. So this is outside facing the cytoplasm, and this is the inside of the endoplasmic reticulum. So N-glycans are already present in the endoplasmic reticulum membrane. And this one is the OST, or the enzyme that transfers N-glycan to newly synthesized proteins. OST is oligosaccharyl transferase. So as the newly synthesized proteins are coming into inside the endoplasmic reticulum, BIP, which is the binding immunoglobulin protein, is present very close to translocon, and it acts as a ratchet and pulls the protein inside the endoplasmic reticulum. And as the protein is getting inside the endoplasmic reticulum, whenever there is asparagine in a specific sequence of amino acids, the N-glycan is conjugated through the action of OST. And once the protein is synthesized, now it is present in this form. It's present in the free form. And now you can see there are these three diamond shapes represent the glucose molecules. The green circles are the mannose residues, and these are the rectangles. Blue rectangles are the N-acetyl glucosamine. So as soon as the protein is fully synthesized, it is inside. BIP is still binding to it to ensure that it does not begin to fold. And while it's bound to BIP, the glucosidase 1 removes the first glucose. Then this glucose 2 removes the second glucose molecules. So now we have a protein with N-glycan having one glucose molecule. In this state, it binds to chaperone proteins, which is calnexin and calreticulin. These two proteins, they recruit PDI, which is the protein disulfide isomerase enzyme. So this enzyme begins to fold the protein into its final conformation. And if there are disulfide bonds in the protein, there should be disulfide. Those disulfide bonds appear at this time. After these disulfide, or the protein is folded properly and disulfide bridges are formed, the glucose 2 again comes in and removes the third glucose molecule from the N-glycan. Lastly, the ER mannosidase 1 acts and removes this mannose residue. Once this mannose residue is removed, the protein is now ready to exit ER, so it will enter the Golgi complex. But what happens if the alpha glucosidase 2 is less because of loss of or egress of alpha subunit of glucosidase 2? So this is this slide I adapted from Herbert and colleague. It shows the same thing, and this part here I showed you how the proteins are synthesized, they get folded, and they get exported out of the ER lumen. But if the proteins are not folded correctly, they enter into the cycle of deglucosylation and reglucosylation. So this cycle goes on for 3 to 4 times. If the proteins do not get folded properly, they bind to BIP, and then they exit out to cytoplasm, where they undergo degradation. But if there is accumulation of protein because of loss of glucosidase 2, it could lead to ER stress. And this has been well documented in an in vivo mouse model, that there is in chronic ethanol exposed mice, there is ER stress. Now, many scientists have worked on this protein synthesis and protein folding in early 50s, 60s, and 70s and 80s, and they found that there is no effect on new protein synthesis in chronic ethanol exposed neurons, and there is no effect on monosidase 1. At that time, they did not know about glucosidase 2 because glucosidase 2 came into picture only in 1996 onwards. 1996 is the first time when glucosidase 2 was identified. So this could impact. We have not done these studies, but we are planning to do. And so this could have an effect and could affect the neuronal physiology because it alters the proteins that are for export or for the cell membrane integration. There are several studies that have shown using specific target proteins that those proteins are less or low abundant in the cell surface of neurons. So from this study, the take-home message is that a simple protein that was originally identified as a substrate of protein kinase C now turned out to be an important protein with multiple functions. And it could also be a potential target of novel therapeutics. So here are some of the information about G2 beta that is known in the literature. It is involved in protein folding of nascent glycoproteins, and we identified it as a RNA binding protein. In cows, it is crucial for follicular development and oocyte maturation. It acts as a calcium sensor protein. It regulates the epithelial calcium channel. It is involved in trafficking of glucose transporter 4 to the cell membrane or the plasma membrane. And incidentally, it is also present on the surface of human sperm, and it is involved with calcium influx, which is very important. This influx is very important for acrosome reaction to take place at the time of fertilization. If there is no calcium influx, then the acrosome activation does not take place. So with this, I think I will end my talk and I will take questions from you.
Thank you, Dr. Meena Kumari. Your talk is very appropriate currently, particularly for India. In India, alcohol drinking is mounting day by day, and new generation are in binge taking of alcohol. So, with this talk, I invite questions from audience.
Okay,
Audience
so
are
how do I?
invited to place questions. Yes. So many questions are not coming. I am certain many questions must be doodling in mind of audience. So I would like to ask one question. I took
Yes.
chance to visit many European countries, and I found many scientists were used to taking alcohol in a gradually in a high amount, but their impairment was quite normal in comparison to India. You have already submitted, you taught us that the range of alcohol consumption in India and the European or the other countries is a wide gap. So, what is the main reason of the upregulation of the genes, why it is not regulated in Indian population by the intake of the high amount of the alcohol?
I think there is some differences with respect to ethnicity, and it depends upon the alcohol dehydrogenase enzyme that is responsible for metabolizing alcohol in the body. So, for example, Asians, I talk about the Chinese people. They have lower amount of expression of alcohol dehydrogenase, so they get very high very quickly in small amount of alcohol. So I do not know if there is any study that has been carried out on Indian population, what are the levels of alcohol dehydrogenase? Because most of the emphasis to date has been mostly on men, European men, population of European men. It's only for the last, I think, maybe less than a decade ago that emphasis was given to females. Initially they thought the woman is not affected, but if you see closely, they also blush. You can see their cheeks are getting red and they're getting hot. So
Okay.
there could be many differences. So one main reason is the expression of the enzyme. And men also get affected, but since they can control their jitters, so you don't see, because you meet them only for a short duration. But if you have to be with them for few hours to few days, then you will realize what symptoms they display. Publicly they control themselves, but uh, in their homes probably they show the effects. But there are definitely, these effects are there.
So it is very interesting to know about the willingness of man. You might be aware to Indian condition. Just few months ago during the lockdown, India has imposed lockdown for a long period, and meanwhile the Indian government was forced to open liquor shop in interest of economy as well in interest of public demand. And it
Yes.
is very discouraging for the country of India, whose economy is based on agriculture, and the liquor consumption is mounting day by day. So government is supposed to open the liquor shop in India. So you can realize in our country the intake of alcohol is increasing, and you aware us about the adverse impact of the alcohol in a several aspect, and new generation faces many challenges in professional as well as in personal life.
Yeah,
So
I think it's very important. See, alcohol addicts, they cannot stop. It is not, even if they want to stop, they cannot stop. So it is not in their control. They can't control it because they don't like the alcohol withdrawal symptoms. So they have to satisfy the glutamate receptors in their brain. Unless they drink it, it's not going to be effective. But younger generation, I would like to say that they should learn a lesson, because these habits devastate families. I mean, it is probably they don't see what is happening to Western world. But once they will see face to face, or they learn more about situation here, then they will realize that we need to stay away from any kind of addiction, whether it is alcohol, cannabis, or opioids, or meth or anything. Crystal meth is now very popular in I think it was in Afghanistan. So India should be very worried, because they are making tons of crystal meth. I just learned few days ago, because my goal is to, I am working on cannabis as well as opioid addiction, because cannabis, we don't know, we don't understand much. Although cannabis is, India has known cannabis, has grown cannabis for a very long time. But it was not so common to consume cannabis. Now, here in the US, it is legal. There are many more countries in the world that are legalizing for both the medical purposes as well as for recreational use, and which is bad, because we do not understand how it impacts our body. And there are certain changes in the brain. I'm working on astrocytes, and I already see how it affects the astrocytes. Astrocytes are the non-neuronal cells.
Ma'am, there are different forms of alcohol like wine, whiskey, red wine, champagne. Has any comparative study that been ever performed to assess which one is more harmful, or whether out of this any one is having a beneficial effect also? Often doctors prescribe brandy or red wine. So has any comparative study ever been done to assess?
When we talk about alcohol, it's the percent of alcohol. See, rum is around 35 to 40%. Vodka, gin, it will have higher amount of alcohol. There are some drinks in China which is 85 to 90% alcohol. Beer has around 4.5% alcohol. So it's I have a slide to show how much alcohol there is in each. I don't know if I can go back and share how much alcohol is in each drink and how quickly it affects the men and women. I had a
But
table.
I have one reservation. It's not that it is the only alcohol percentage. Had it been the case, then it was very easy to prepare 10% alcohol by adding water, 20% alcohol, and make whiskey rum. There are other chemicals also which contribute to the properties of red wine or brandy or
Red
whiskey, other
wine
than the alcohol.
Red wine has polyphenols, which is antioxidant, and that's why it has cardiovascular beneficial effects to cardiovascular. It specifically affects the lining of the blood vessels, which is the endothelial cells.
So, is it that all the alcohols are harmful to health, or even some have
Mhm.
beneficial property also? Or
All
any
al-
study which has been performed extensively to examine all these aspects? Whether there is some beneficial aspect also associated with alcoholism, other than these harmful ones that we always talk about?
uh, there is beneficial effects of alcohol, and that's red wine. And red wine is because of polyphenols that is contained in the red wine.
Often in
And
Indian context,
those uh, those polyphenols are surprisingly they're also present in peanuts.
often in Indian
So,
context, we have seen that doctors often prescribe brandy, a little amount of brandy to certain persons who are having certain disorders. So even sometimes
I'm,
doctors themselves prescribe.
I am not aware of that. I mean, I lived at least 22 years in India, but I did not see. Maybe those were days or previous days when I did not experience that.
Okay,
I
madam.
mean, I
Thank you,
didn't
ma'am.
see, but I'm surprised that they are insulting people.
Okay, ma'am. Thank you, ma'am.
Oh, you're
So,
welcome.
thank you so much, madam, for this wonderful lecture. I wish you see you in many more academic interactions offline as well as online. So, personally, we
Thank
are
you.
thankful to you.
Thank you. Bye
So,
bye now.
thank you so much. So, I am inviting next speaker, Dr. Deobrat. I think he is connected to the conference, and he is supposed to deliver his talk on Epitranscriptomics, a new RNA code for the regulation of gene expression. Dr. Dixit is working in states, and fortunately, he is native of India. So, naturally, we have a different kind of bonding with Dr. Dixit. Dr. Dixit is having a wonderful resume, and organizing secretary has placed over the table. Dr. Dixit is a postdoctoral associate in the laboratory of Dr. Robert Wessler Rea at the Sanford Burnham Prebys Medical Discovery Institute, California, United States of America. Before joining Dr. Wessler Rea in 2020, Deobrat worked in the laboratory of Dr. Jeremiah Rich in the University of California, San Diego, United States. Dr. Dixit did his PhD in neuroscience under the supervision of Dr. Professor Alora Sen in National Brain Research Centre, New Delhi, India, and he took duration from 2007 to 2014, about 77 years. After finishing his master's from the Department of Zoology, Banaras Hindu University, Varanasi, for 2 years from 2005 to 2007. He published more than 30 papers in his account in the area of neuro-oncology and development neuroscience. His prestigious publication occupies the word Nature Journal, and he deserves for a lot of appreciation for getting this chance to publish his finding in Nature. And apart from that, he has published his discoveries in cancer discovery, cell death and disease, genes and development. And these are the limited number of the journals, those names here I am taking. And he has 1400 citation to his academic account. Dr. Dixit is a current area research is to study the molecular and cellular mechanism of glioblastoma. With this short biodata, I am inviting Dr. Dixit for the talk. Now it is over to Dr. Dixit.
Hi, a very good morning to all of you. I'm pleased to be here, and it's an honor to present in front of my seniors and teachers, some of my teachers. So I will share my screen now.
And a very good morning to you also. We are keen to listen to you.
Okay, it's not letting me share.
Yes, you take your own time. We are with you.
Is my screen visible now?
Yes, it is in the process. Now it is available. You can proceed now, please.
Perfect. So,
Dr. Dixit, please click on the hide button also beside stop sharing.
okay. Thank you. So, I think I am the odd one here. My work is very little related with the basic neuroscience. I work on brain cancers. And specifically, I use brain cancers as a model to study a new facet of gene expression regulation, which is Epitranscriptomics, where we deal with the various modifications that happen in RNA molecules. So first, I will introduce briefly to the model system that we work on, that is glioblastoma. Glioblastoma, as we see, accounts for almost 28% of all CNS malignancies that arise, and its origin and spread is diverse. As we can see in these MRI scans, it can originate in different shapes in different regions of the brain. In some cases, it will show up as a small tumor, but in a very important brain region. And if not treated, I mean, there is no treatment, but if not managed, it grows rapidly. And it accounts for more than 80% of all the cell deaths caused by CNS malignancies. So 28% of tumors are causing 80% of cell deaths, 80% of patient death. So it's characterized as one of the most lethal of all the cancers. And the mean survival of a patient who is diagnosed with glioblastoma right now is 15 months, which is pretty sad. Extensive amount of work has been done, but still, as we can see, the success is very limited. We can have a basic idea of how much progress we have done. In 1900s, 120 years back, if a patient walked into the clinic with a high-grade glioblastoma, his life expectancy was 11 months. And now we have 15 months. So basically, in 120 years of research, we have given 4 months to our patients. So that tells that amount of work is still needed to be done to manage these deadly tumors. In summary, I will say just that glioblastoma is bad. And during years of research, we have identified the key region behind the aggressive nature of these tumors. That is the presence of a very small fraction of cells, which are known as tumor stem cells or glioblastoma stem cells. So these stem cell populations, the glioblastoma is a heterogeneous tumor, and these glioblastoma stem cells sit at the apex of the hierarchy. in the heterogeneous cell population. And my lab and several other labs have characterized these hierarchical cell populations, and we have characterized them by their different abilities to generate tumor in mouse models. So stem cells can be found in two different cellular stages in a tumor. They might be quiescent. In a healthy growing tumor, the stem cells are usually in a quiescent form where they are not dividing, they are just sitting in the tumor. But once these tumors are faced with the microenvironmental challenges like chemotherapy or radiation, these glioblastoma stem cells, I will call them GSCs for throughout this presentation. So these GSCs, they start rapidly proliferating and they can give rise to a whole new tumor in less than 2 to 3 months. The various studies have established the role of GSC in several aspects of tumor growth and maintenance. Some of them I have summarized in this figure. Okay, a quick question. Is my cursor working as a pointer?
Yes, of course.
So when I'm moving my cursor, it's showing up in the presentation, right? Cool. So this slide summarizes the various features of the glioblastoma stem cells that has been studied, and inhibitors has been developed for these features, like the role of glioblastoma stem cells in epigenetic regulations, where BMI1 and EJTS2, as well as TERT inhibitors have been in clinical trials. Similarly, GSCs regulate metabolic dependencies of tumors, immune therapy, and various microenvironmental factors, as well as post-transcriptional regulation of gene expression. The post-transcriptional regulation is my area of interest, and I will elaborate on it in my presentation. So, going back to the basics of gene expression, the basic central dogma that is taught in schools and colleges, proposed by Francis Crick in 1948. It's a process by which the genetic instructions in DNA are converted into functional products that are proteins in a cell. So basically, a DNA undergoes replication to make its own copies. Then it gets transcribed into RNA, and RNA gets translated into proteins with the help of ribosomes. When further studied, this process leads to the complexity of the gene regulation, where we know that the DNA which is in the cell in a condensed form in the form of chromosomes. The DNA fibers are packed into chromosomes with the help of proteins called histone proteins. The first level of regulation arises where DNA itself gets modified mainly by methylation. Then histone proteins, which are packing DNA, they gets modified. There's a whole battery of modifications are reported for histones, which determine open versus closed conformation for chromatin and regulate the transcription of DNA. Then there are regulations at the level of transcription and post-transcriptional regulations. When we were in the classrooms doing our college degrees, post-transcriptional regulations were limited to 5' capping, poly-A tailing, and alternative splicing. And then there was regulation on the basis of at the level of protein translation and post-translational modification of proteins. recently, a new level of regulation has been added, which is technically a post-transcriptional regulation, but this is the identification of the modifications in RNA moieties. The RNA M6A modification, which was first reported in 1974, but after that report, the functions were not further elucidated, and its role was started to get appreciated when 2012 this Nature paper came out, where they came up with a technique to profile the global M6A levels in a cell. So I'll summarize the modification and its regulators in brief. So like any other modification of DNA or protein, RNA modifications are also governed by three different group of proteins. There are writer proteins or methyltransferases, which install modification on an RNA. Then there are erasers or demethylases, which remove modifications from RNA species. And then there are reader proteins, which are largely known as class of RNA binding proteins. These proteins, they read the methylation code on RNA and lead to further signal transduction in the cell. The methylated RNA basically leads, the methylation basically governs the cellular export of RNA, and then based on the type of reader protein or RNA binding protein bound to the modification, it may lead to mRNA decay or its stability and translation. In past 8 or 9 years, both oncogenic and tumor suppressive role of these modifications has been proposed. So basically functions are pretty complex, and we are still in the initial phase of understanding the role of RNA modification, specifically in cancers. So this is the summary of what I just said. So now I'll dive into my work. So we decided, when I joined Dr. Jeremy Rich's lab, there was literally no literature on RNA modification in CNS malignancies, and we decided to adopt the protocol published in 2012 and profile M6A modification in glioblastoma stem cells, and we chose neural stem cells as our controls. So this is the brief summary of protocol. Basically, we grow cells in a monolayer, then we collect them, isolate RNA, and perform a pull down using a specific M6A antibody. After the pull down, this process pulls the all modified mRNA, then we subject it to high throughput sequencing, and then using bioinformatical pipelines, we are able to characterize the presence of modification at single nucleotide level. So when we compared the M6A profile of glioblastoma stem cells with neural stem cells, I'm writing neural stem cells as NSCs and glioblastoma stem cells as GSCs. So when we compared NSCs to GSCs, we found that the whole battery of mRNA are differentially modified in both cell populations. But to our surprise, the genes which form a stem cell signature and are essential for stemness and tumorigenicity of glioblastoma were heavily modified with this modification. So some small examples are OLIG2 and MYC. And then we looked for the M6A modified genes into the patient data sets and how many of these are essential for tumor growth in human patients. And we found a whole bunch of them were characterized as essential genes for glioblastoma. So all we can say from here that M6A modification tags important oncogenes in glioblastoma stem cells. Then we decided to gain some insight into the role of this modification on gene expression profile of the modified genes. And what we found that most of these genes which are modified with M6A were heavily overexpressed in the tumors, both patient tumors and glioblastoma stem cells. So this panel shows that genes with glioblastoma stem cell specific peaks are overexpressed, whereas genes with neural stem cell specific M6A peaks, which are unmethylated in glioblastoma, are downregulated in patient tumors. Similarly, we mined a gene expression data set, large gene expression data set of glioblastoma patients, and we observed that this is a data set of more than 1,082 patients, and in these patients, the M6A modified genes that we identified in our glioblastoma stem cells, these genes are overexpressed in patient tumors when compared to neighboring non-tumor tissue. This is a simple example. We took MIC as an example, and we see large M6A peaks in our sequencing data in our glioblastoma stem cell models compared to neural stem cells. And of course, for example, MIC expression is very high in glioblastoma stem cells and in glioblastoma tumors. So the modification leads to increased expression. That's the initial indication here. Similarly, we explored the expression of genes which are not modified in glioblastoma stem cells and are heavily modified in neural stem cells. These genes are downregulated in glioblastoma tumors when compared to neural stem cells. We take a MN1, it's called meningioma 1, and it's a known and highly effective tumor suppressor in glioblastoma stem cells, and we see that a tumor suppressor gene which is downregulated in tumors is not modified. So this is our first conclusion from our first experiment, and the further and the downstream analysis that RNA M6A peaks mark the potentially and potentially regulate critical cancer specific genes in glioblastoma in a location and cell type specific manner. I'm not showing the data. We analyzed the location of M6A marks in a transcript, and we find that presence of M6A on 5' regions of RNA is specifically correlated with the higher expression of these transcripts. So now we know that modification is important for glioblastoma stem cells. So we decided to explore the role of different regulators of modification. So as I showed in my introductory slide, there are writers, readers, and erasers of the modification. So on this slide, on this first graph, we can see that the expression of two known writer proteins, metal-3, metal-14, two eraser proteins, ARBH5, FTO, and the 5 YTH family RNA binding proteins, which are the reader proteins. And when we explored their expression in tumors, we find that the reader protein YTHDF2 is overexpressed in high-grade glioblastoma. We further explored the expression of these genes using Western blotting and compared them with neural stem cells, and we again see that YTHDF2 is selectively highly expressed in glioblastoma stem cells, along with the writer protein metal-14. This is the quantitation of Western blots showing that YTHDF2 and metal-14 are significantly overexpressed in glioblastoma stem cells. To further establish the role of these overexpressed proteins, we thought that writer protein is more important. So we first knocked down the metal 14 in glioblastoma stem cells, and we see a decrease in cell viability when we knock down metal 14 in cell culture. But here is a Kaplan-Meier curve showing the effect of metal 14 knockdown in mouse models, and we don't see any effect of knockout in growth of glioblastoma tumors in mice. So I'll summarize. I will briefly tell about the in vivo mouse experiments that we perform. We take the glioblastoma stem cells, genetically manipulate for our desired target, and implant them in the brain of immunocompromised mice. And then we monitor tumor growth and the survival of mice. So these Kaplan-Meier curves, they will come throughout my presentation, and they represent the survival of mice bearing either control or genetically manipulated cells, here for knockout of metal 14. And as we see that the mouse with either control cells or metal 14 knockout cells, they all show similar survival, which means the tumor growth is not altered between different groups. That indicates that metal 14 doesn't affect the tumor growth in mouse models. So then we moved ahead to YTS proteins. So YTSDF2 family of proteins actually comprises of 3 proteins: YTSDF1, YTSDF2, and YTSDF3. All these 3 proteins have been reported to have functional redundancy, and they perform similar functions. Till the reports, when we were working on this project, that was the report, the literature report. So we decided to knock out all 3 of them, since although we found overexpression of only YTSDF2, but YTSDF1 and YTSDF3 knockout had no effect on cell viability. But the overexpressed protein YTSDF2, when we knock it out using CRISPR-Cas9 system, we see a massive decrease in the cell viability of glioblastoma stem cells. Also, we performed the stemness assays. The gold standard is in vitro limiting dilution assay, in which we quantify the stem cell frequency in a cell population in a culture dish, and we see that stem cell frequency of this cell population gets heavily reduced when we knock out YTSDF2. Similarly, their sphere formation ability is reduced. So YTSDF2 is affecting both the viability and stemness phenotype of glioblastoma stem cells. To establish its role, we further overexpressed knockout resistant YTSDF2, and we see that decrease in cell viability caused by YTSDF2 knockout can be rescued by exogenous overexpression of the protein. This establishes that YTSDF2 is necessary and sufficient for the survival of glioblastoma stem cells. We further performed in vivo tumorigenesis experiments in the mouse, and as we see with these hematoxylin eosin stained brain sections and with these Kaplan-Meier curves, that YTSDF2 knockout heavily retards the tumor growth in mouse, and it increases the survival of mouse bearing tumors. So definitely affects the tumor growth. This is analysis of, and this is a bioinformatic analysis of the patient tumor data set, and we wanted to establish or at least give an indication of the functions of YTSDF2 in other cancers. So Broad Institute has a larger data set where they have performed CRISPR screens, whole genome CRISPR screens, in roughly a little bit more than 4,000 tumor cell lines, and they have established dependency scores for every gene. And if anything which is in negative is considered a dependency. As we see in all these different cancer forms that the data set has data for, a YTSDF2 knockout leads to negative dependency, which means that all these tumors depend on YTSDF2 for their survival. But we, in this presentation, will keep our focus on glioblastoma and glioblastoma stem cells. So concluding till now, the second conclusion that we add, that YTSDF2 is a specific and potent regulator of glioblastoma stem cell viability and their stemness potential, and regulates their tumor formation ability. Now we wanted to characterize the targets, which the downstream targets which are regulated by YTSDF2 and the M6A modification. For this, first, we again utilized the online data sets which are available from patient tumors, and we did bioinformatic analysis to characterize the genes whose expressions are positively correlated with YTSDF2 in patient tumors. Again, we are dealing with slightly more than 1,000 patient tumor data set here. And we see that the genes which are positively correlated with YTSDF2 expression are part of RNA metabolism and protein synthesis pathway, and are also related with RNA splicing and transcription, which is not surprising because it's we are dealing with RNA modification pathway, so it affects the genes, primarily affects the pathway along with some glucose metabolism. When we looked into the specific targets, then we see that the MYC, which was highly M6A modified, the MYC pathway and its targets are down-regulated, are positively correlated with YTSDF2. To further identify the YTSDF2 targets in our glioblastoma stem cells, we performed RNA sequencing in YTSDF2 depleted glioblastoma stem cells, and we find that when we knock out YTSDF2, there are 512 genes which are down-regulated, whereas there are 498 genes which are up-regulated in glioblastoma cells. The down-regulated genes again consist MYC and several IGF binding proteins, and VEGF-A, which is a core regulator of vascularization in tumors. Similarly, we see a decrease in MYC targets and ribosomal and translational and RNA processing pathways upon YTSDF2 knockout. So by RNA sequencing, we are not only able to identify the direct targets, we are also able to identify the pathways that are being affected in glioblastoma stem cells. Now we co-analyzed our RNA sequencing data along with our M6A profiling data. That was my first figure. And we overlapped the two data sets to see what is the trend of gene expression among M6A modified transcripts in the cell. And what we observed that the genes which were modified with M6A, when we knock down YTSDF2, they are down-regulated, whereas the genes which have no M6A peaks, they are upregulated. So that tells that, at least gives us a primary indication that M6A tagged genes are maintained in a steady state transcriptional level by YTSDF2. Now we wanted to elaborate further and identify the direct binding of YTSDF2 to the mRNAs. For this, we employed a technique called CLIP, which is again a modification of the first technique that we used. Here we, in place of using M6A antibody, will use direct YTSDF2 antibody and pull down the RNAs that are bind to YTSDF2. So this is initial results, and comparing YTSDF2 binding targets in neural stem cells that are HNP1 and NSC11 versus glioblastoma stem cells. The two lines that we used are called 412 and 387. As we see that YTSDF2, we observed that it was overexpressed in glioblastoma stem cells, and it's also showing massive increased RNA binding activity in glioblastoma stem cells when we compare with neural stem cells. The RNA binding activity was so limited in one of the two neural stem cell lines that we were not able to get enough transcripts to sequence. So basically, we pull down the RNAs bind to YTSDF2, and then we again subject to high throughput next generation sequencing. And that identifies the mRNAs which are directly bound by YTSDF2. So just I will summarize this dense slide by putting the larger picture. So this Venn diagram simply shows the overlap of genes which were down regulated upon YTSDF2 knockdown, and which had both M6A and YTSDF2 clip peaks, which means they are modified with M6A and they are also bound by YTSDF2 directly. So we overlap the genes which are M6A modified, they bind to YTSDF2 directly, and to the data set where we identify the genes which are down regulated upon YTSDF2 knockout. So overlapping the three different next generation sequencing data sets, we can zero down to 30 genes. These 30 genes are M6A tagged in glioblastoma stem cells. They are bound directly by YTSDF2 as a RNA binding protein. And when we knock out YTSDF2, their expression goes down. Among these 30 selected genes, we thought that, okay, these are the direct targets of YTSDF2, direct oncogenes. And we find MYC and VEGF-A, two of the most important oncogenes in almost all cancers. They fall into this small gene list. So until now, we are able to identify the direct M6A and YTSDF2 targets in glioblastoma stem cells. Then we performed qPCR analysis to confirm our next generation sequencing high throughput experiments, and we picked 7 of these 30 genes, and we checked their levels using qPCR, and we find that we are able to confirm our findings that YTSDF2 knockout leads to decrease in their mRNA levels. Now, the question was, what is the method of overexpression or the stability of expression by M6A modification and YTSDF2, and whether presence of M6A really plays a role in it. So we first again used YTSDF2 specific antibody to pull down 2 of the important targets, MYC and VEGF-A, between NSCs and GSCs using RNA immunoprecipitation, and we identify specific binding of YTSDF2 to MYC and VEGF-A in glioblastoma stem cells only. And then we quantified the half-life of these mRNAs in control versus YTSDF2 knockout conditions. And we see that stability of MYC and VEGF-A mRNAs are heavily affected when we knock out YTHDF2. So YTHDF2 stabilizes these mRNAs. Now we wanted to establish the role of M6A in the stabilizing process of these mRNAs. For this, we took two approaches, one indirect and one direct approach. In an indirect approach, we knock down metal 3, which is a writer of M6A modification. So the hypothesis was that when we knock out the writer or methyltransferase, the M6A modification will be removed from its target genes. And as we see that knockout of methyltransferase metal 3 leads to massive global decrease in M6A modification as quantified by ELISA. When we knock out metal 3, we see the binding of YTHDF2 to MYC is decreased many folds. So that indicates that once we remove M6A from the MYC, its binding to YTHDF2 is affected or downregulated. That establishes that probably presence of M6A is important for the binding of mRNAs to YTHDF2. Now, we took a direct approach to establish the role of M6A in YTHDF2 functions, for which we generated the M6A binding mutant forms of YTHDF2 protein. We generated two mutant forms where mutations were created in the M6A binding pocket of YTHDF2. And as we see that these M6A binding mutant YTHDF2 proteins, they lose their binding affinity to MYC in both the glioblastoma stem cell lines. Now we wanted to establish whether the M6A binding function of YTHDF2 actually regulates its role in the cell survival, for which we again performed the rescue experiment with wild type and M6A binding mutant form of YTHDF2. As we see that knockout of wild type YTHDF2 leads to decrease in the cell viability. Knockout of YTHDF2 leads to decrease in cell viability. But when we overexpress wild type form of YTHDF2, we see the rescue of viability, but the M6A binding mutant forms are unable to rescue the viability in YTHDF2 knockout cells. This establishes that the M6A binding function of YTHDF2 is important for its oncogenic role in glioblastoma stem cells. And its specific binding to MYC is affected by presence of M6A on MYC mRNA. So these were kind of mechanistic experiments. Concluding these mechanistic experiments in brief, we find that YTHDF2 stabilizes MYC and other important oncogenic transcripts in glioblastoma stem cells. And effect of YTHDF2 on transcript stability is dependent on presence of M6A modification. So these experiments establish both the importance of M6A modification on oncogenic mRNAs and the downstream signal transduction by its binding protein YTHDF2, which regulates the stability and tumorigenicity of glioblastoma stem cells. Now we moved ahead and decided to establish the downstream targets of YTHDF2. The idea was to find, since M6A pathway and YTHDF2 has no known drugs till date, M6A inhibitor was reported like 2 months back from a group in London, and there is no inhibitors for any of the M6A binding proteins. So we wanted to explore the downstream targets so that we can find something druggable. Actually, the idea of the whole work is to find something which can be useful in clinic. So since MYC, we identify MYC as a direct downstream target of YTSDF2, we wanted to establish its role in YTSDF2 functions, and as we see that decrease in cell viability upon YTSDF2 knockout can be rescued by MYC overexpression, and similarly MYC knockdown affects the expression of all the YTSDF2 target genes that we have characterized. So this and the gene expression changes in YTSDF2 knockout cells can be rescued by MYC overexpression. So these experiments establish MYC as a direct downstream target of YTSDF2 and responsible for its oncogenic functions in glioblastoma stem cells. But unfortunately, with all the efforts in past 40-50 years of its discovery, MYC is still not druggable. There is no strategy to target MYC in cancer cells, and millions and millions of money, dollars, and a lot of groups, including our group, is putting constant effort to identify a way to make MYC druggable. But since it's not druggable yet, we went ahead and explored other targets which might be downstream to YTSDF2 MYC axis. Okay, sorry. Before that, we explored the role of YTSDF2 and in MYC stability and compared it between neural stem cells and glioblastoma stem cells, since we find that YTSDF2 knockout doesn't affect the viability of glioblastoma neural stem cells, whereas we have seen that it affects the viability of glioblastoma stem cells. So we find that the stability of MYC is affected by YTSDF2 only in glioblastoma stem cells, and MYC mRNA levels are not affected by YTSDF2 in neural stem cells. We identify both on the mRNA stability and protein expression levels. So this establishes the selectivity of YTSDF2 targeting, and it doesn't affect the normal cell population of brain, which will be really useful because it will provide a better therapeutic index if we are able to target YTSDF2 in a patient. So concluding till now, we are adding another conclusion now that YTSDF2 represents a GSC-specific dependency that supports glioblastoma growth through GSC-specific stabilization of MYC transcripts. Moving ahead, as I said, we wanted to identify something druggable. So we looked into our RNA sequencing data set, and one of the most highly downregulated gene was IGF-BP3, IGF binding protein 3. Actually, the whole IGF-1 pathway was downregulated upon YTSDF2 knockout, and IGF-BP3 was top downregulated genes among the downregulated genes. So we first explored the binding of YTSDF2 and the transcript stability in YTSDF2 knockout cells, and we identify binding of YTSDF2 in both neural stem cells and glioblastoma stem cells, although a significantly higher binding in glioblastoma stem cells of YTSDF2 on IGF-BP3 mRNA, but we don't see any effect on its transcript stability upon YTSDF2 knockout. So we reasoned that since the transcript stability is not affected, probably IGF-BP3 is not a direct target of YTSDF2, and it might be affected by MYC as a downstream target. So first we wanted to establish the role of IGF-BP3, and we found that IGF-BP3 knockout heavily affects the cell viability and stemness of glioblastoma stem cells. We explored the patient data sets, and we find that IGF-BP3 is heavily overexpressed, and it affects the survival of human patients, where presence of high IGF-BP3 expression leads to poor survival in glioblastoma patients. In our experiments also, we find that IGF-BP3 knockout significantly affects the tumor growth in mouse model of glioblastoma, with the increased survival of mice with IGF-BP3 knockdown tumors. So this establishes that IGF-BP3 is an important target, which is downstream of M6A YTSDF2 pathway. Now we wanted to establish it as a downstream target of YTSDF2, so we overexpress IGF-BP3 in YTSDF2 knockout cells, and we see that it is able to partially rescue the cell viability of YTSDF2 knockout cells. Similarly, it is able to rescue the tumor growth in YTSDF2 knockout cells when overexpressed in vivo. The tumors, the YTSDF2 knockout cells hardly form any tumor in mice, but when we overexpress IGF-BP3 in YTSDF2 knockout cells, the tumor growth is restored. That establishes IGF-BP3 as a specific downstream target of YTSDF2. Now, we explored the druggability of the pathway, and for this, we again utilized a Broad Institute high throughput data set. So it's called Cancer Therapeutics Research Portal. What Broad Institute researchers has done, that they have explored the effect of more than 5,000 FDA-approved drugs on a whole range of cancer cells in vitro. And they performed RNA sequencing at the same time of drug treatment. And then they have generated a high throughput data set. So how we can utilize, so we decided to smartly utilize this data set by creating a correlation with the YTSDF2 expression with the drug response to different drugs. So this graph, as we can see, it depicts the therapeutic compounds which show some perturbation in YTSDF2 differentially expressing cells. And the drugs on positive axis, y-axis, are the compounds towards which YTSDF2 expression causes resistance. So if a cell is expressing YTSDF2 as high level, though those cells will be resistant to these drugs, whereas the high YTSDF2 expressing cells are supposed to be sensitive to these drugs. We had a whole list of FDA-approved drugs here, which were predicted to sensitize cells with high YTSDF2 expression. We chose one of them, which is lincitinib, because lincitinib is a specific IGF-1 binding protein inhibitor. So this also not only predicts drugs for us, it also provides us a mechanistic validation of our work, where we have established that M6A pathway in glioblastoma operates via YTSDF2 IGF-BP3 axis, and a larger data set is predicting IGF-BP3 inhibitor as a specific target for YTSDF2 overexpressing cells. So we decided to explore lincitinib as an anti-glioma drug. And for in vitro experiments, we utilized 6 of glioblastoma stem cell lines and 3 of neural stem cell lines. And to our sweet surprise, the high dose of lincitinib treatment still has no effect on the survival of neural stem cells. So neural stem cells, which express significantly low levels of YTSDF2, they are not sensitive to lincitinib treatment. Whereas glioblastoma stem cells, which express high levels of YTSDF2, they are sensitive to sub-micromolar doses of lensitinib. This was kind of, we were really happy to see these results, because again, it is giving us a broad therapeutic index where probably normal cells of brain will not be affected by lensitinib, whereas it can specifically target glioblastoma cells. Again, we performed in vivo experiments in mouse models using lensitinib treatment, and we see that lensitinib treatment has retarded tumor growth in mice models bearing tumors generated by glioblastoma stem cells, and it prolongs the survival of drug-treated mice. So, summarizing the whole story, what we found in this study, that RNA M6A peaks mark and potentially regulate critical cancer-specific genes in glioblastoma in a location and cell type specific manner. YTSDF2, the M6A reader, is a specific and potent regulator of glioblastoma stem cell viability and stemness. Then we characterized the functions of YTSDF2, and we found that YTSDF2 specifically stabilizes MYC and other important oncogenes like VEGF-A at mRNA level in glioblastoma stem cells. And this effect of YTSDF2 and on stability of its target mRNAs are dependent on M6A modification. And YTSDF2 functions are GSC specific, and it doesn't affect neural stem cells viability, and its stabilizing effects on MYC are specific to glioblastoma stem cell population only. Then we identified IGF-BP3 as a downstream target of YTSDF2 MYC axis, and we found that IGF-1, IGF-1R, IGF-BP3 pathway inhibitor lensitinib can retard tumor growth by oral administration in the mouse models of glioblastoma. So here is a model, brief model of the work. We were able to publish this work in Cancer Discovery earlier this year. Actually, the paper came online in January 2021. So with this, I will conclude my presentation. And okay, I'm somehow not able to find my thank you slide, but I would like to thank organizing committee again for inviting me for this presentation. And with this, I'm open for the questions.
So, Dr. Dixit, you have given a wonderful talk. Before placing my question, I would like to request audience to place questions.
Dr. Dixit, congratulations to you and your research team for adding a new dimension to cancer research. I have got a number of questions.
Yeah. Sure.
You have talked about METL, RNA, M6A, YTH, EDF2 family.
Yeah.
Do they play a role in other types of cancer? Or
yeah, now
for glioblastoma?
No, in last 3 years there have been more than hundreds of publications. So now people have characterized their roles in at least the prominent cancers, like all the blood malignancies. Actually, a paper came right before my paper, which technically scooped my work a little bit, where they showed that YTSDF2 regulates the AML stem cell population specifically without affecting the differentiated blood cancer cells. And then there has been reports in what I can recall, at least in lung cancer, gastric tumors, and and kidney cancer. So there have been several reports by now. In past 2 years, there has been so many reports.
The second question is that is Y2 SDF2 is it constitutively expressed in other cell type also, or is it there is some special cell types in which it is expressed?
We see its lower expression in glioblastoma stem cells.
What about
A
other
lower
tissues?
expression in neural stem cells. We have not
What about
explored the
yeah,
expression.
any report from other groups like
But if
whether
you
it
look
is
at
specific
this,
to
say this
liver
broad,
or lung?
yeah. So if you look at the Broad Institute data set, which is a CRISPR screen data, and they performed RNA sequencing on these tumors also. So at least they have detected expression in all these tumor types. They don't have any control cells here.
Okay. And does there
So if we
exist
look at,
any? You continue.
yeah, there is I see fibroblasts as their controls, where but other than that, they have all these cell types where they have detected expression and they have performed a CRISPR screen, large scale CRISPR screen, where they are able to knock out and see the effect on viability.
You are talking about Y2S2 DF2 has got a role in
YTSDF2. Yes.
multi-drug resistant protein. Does there exist any crosstalk with other multi-drug resistant proteins like P glycoprotein, MDR1, BCRP, etc.?
No, I'm not talking about drug resistance here. This is basically the effect on viability of these tumors. And
Okay.
that's a very interesting question. I have not seen any report of YTSDF2 and its association with drug resistance or drug efflux. Not that I can recall.
Okay. And you talked about linestininib. Is it an approved drug for human use?
Yeah, it has been in clinic for a while and used. I think it has been used for, if I remember correctly, rheumatoid arthritis and few other minor things. So lincitinib as a drug is in market for since 1970s, but it is not implicated in cancers.
Okay. And the last question, all the parameters that you have talked, METL, YTS due to RNA M6A, at the downstream level, do they play any role in regulation of enzymes associated with cancer, disease, development or progression, or maybe reprogramming of energy metabolism?
Yes. So since I was already beyond my timing, so I didn't go to future directions. So right now we are exploring the role of M6A modification in metabolism. And and how we decided to do it, if you look here, our RNA sequencing shows that apart from RNA metabolism and protein synthesis, other pathways which are affected are TCA cycle and glucose metabolism.
Okay.
Right now we have a kind of pretty mature story where we see, I can give a little bit of information where we see that malate aspartate subtle largely regulates M6A modifications, and the methyl donor metabolism has important role in the global modification. So we are looking at the metabolic regulation of RNA modification right now. We have a fairly mature manuscript which should be out, if everything goes well, in few months.
Okay. Thank you,
And we
Dr.
have
Dixit.
identified a specific metabolic gene which regulates which impacts global M6A modification and the binding of RNA binding proteins to M6A.
Thank you and best wishes for your new upcoming paper. Thank you.
Thank you, Via.
So, Dr. Dixit, you are already snowed over with a series of questions by Dr. Kumar. And it is very true that the longevity of the cancer patients have been increased by the scientists, by the hard work, the scientists like you who works days and night. And making the drug availability is not a easy task, it's a Herculean task, and it takes time. I have only a small question because time doesn't permit. That knocking out the YTS DF1 and DF2 may affect the vital activity of different organs, because it has been seen that the drug impact of the cancer affects different organs, specifically liver. So, is there a probability of the same happening by
Ma'am, we
this
don't
knocking
have
out?
we have knockout mouse model for the metal 3 and metal 14.
Okay.
And
So, is
they survive and they grow, they like survive, and with minimal like we at least metal 14 knockout mice has shown some neurological defects, but metal 3 knockout mice survives fine.
Okay, so it's a very
So
good thing.
yeah, we don't have knockout mice for any of the YTH proteins yet. So, but
Okay.
yeah, the M6 methyl transferases, their knockout mice grow into adult mice and they survive.
It's a very great thing. So we wish you for the drug availability for this particular cancer cell.
Thank you,
So
ma'am.
thank you so much, Dr. Dixit. I wish you for your best success and would like to see you in many more academic instructions offline and online. Thank you so much.
Oh, thank you very much. I mean, I can't say how happy I am after this presentation. I don't know how I did. I'm gonna torture Rajkumar Bhaiya to tell me what errors I have made. So, Dr. Rajkumar Koiri is my senior, from VHU days. So,
Okay,
uh,
always there is a
he's my
scope
first
of.
mentor, research mentor. So yeah, but this was an immense honor to present in front of the audience and the organizing committee at Sagar University, and I'm really
And
happy and proud of to be able to present here.
And you
Thank
had
you.
a wonderful presentation. It was very lucid and make us understandable. Thank you so much.
Thank you.
So I am inviting the next eminent speaker, Dr. Prashant Kumar Modi, who has already joined us. He is from Center for System Biology and Molecular Medicine, Yenepoya Research Centre, Mangalore. He has a wonderful resume. Dr. Modi is currently working as assistant professor at Yenepoya University, Mangalore. I am sorry for mispronouncing this Kannada word Yenepoya if I am not pronouncing correct. He obtained his master's degree in biochemistry from the School of Biochemistry, Devi Ahila University, Indore, from our own state. And after that, he pursued his doctoral degree from BHU Varanasi under the mentorship of late Professor M.S. Kaungo on expression and regulation of glial specific genes in the brain of mice as a function of ACE. After his PhD, he joined Dr. Pushkar Sharma's laboratory at the National Institute of Immunology, New Delhi. For his postdoctoral training and later joined Yenepoya University in 2016 as assistant professor. He is the recipient of several national awards like the DBT Research Associates from the Department of Biotechnology, Government of India, Best Oral Presentation Awards in 27th Annual Meeting for the Society of Neurochemistry India and International Conference on Recent Advances in Molecular Mechanism in Neurological Disorder held in AIIMS, New Delhi. His current research work includes characterization of novel signaling pathways in neurodegenerative disorders. He is also interested in dissecting the molecular mechanism involved in neuroprotective functions of Ayurvedic medicines. He has published his research findings in several international journals of high repute, like Cellular and Molecular Biology of the Cell, and Cellular and Molecular Neurobiology, Journal of Cell Communication and Signaling, Journal of Ethnopharmacology. He has produced one PhD student in a short span of his career. Currently, three PhD scholars are working under his leadership. He is a life member of several scientific societies like Association of Gerontology and the Indian Academy of Neuroscience in India. He is on the editorial board of reputed journals, including Evidence Based Complementary and Alternative Medicine Cell Biology International. In the short span of his career, he has done an exemplary work for the science and contributed new things to the area. Now I am inviting to Dr. Modi for his wonderful talk on application on multi-omics technology in deciphering brain functions and disease. In the time of the current scenario, his job may be a wonderful because in current situation we are not talking towards one technology. So now it is over to Dr. Modi for the talk. Dr. Modi, yes, please. Please unmute yourself. I am not getting your voice. May I request you to please unmute? Could you please unmute yourself? You are not audible. In the bottom panel of the screen, there is mute and unmute. I think that mistakenly you have muted. Dr. Rajkumar, can you approach online?
I think Dr. Modi is in unmute mode, but I think there is some network connectivity issue from his side. I think Dr. Prashant, you can disconnect yourself and you can rejoin. I think that will solve the problem.
You can also talk over the phone to give him the solution. Probably he might not getting our voice because I am not getting any gesture from Dr. Modi. Dr. Rajkumar, please talk over the phone. I think his voice is not coming and he is also not receiving.
He's connecting. He's having
Okay.
some issue. He's joining.
Okay, thank you. It happens to best of us, so no problem. Meanwhile, he's trying to connect. I can see that Dr. Rajendra is here, and may I know from where you are, Dr. Rajendra? So this session was very interesting and we had very good speakers during
Dr.
this
Prashant,
session.
I think Dr. Prashant has joined.
Okay,
Now,
okay.
now I am audible.
That's perfect. That's perfect.
Actually, I'm extremely sorry for the delay, and I'm thankful for the kind introduction, and
And
I'm also thankful to the organizer for giving me the opportunity to present my work in this conference, online webinar, and I'm now sharing my slides.
no issue, Dr. Modi. It happens to rest of us. So this is the platform of online and we face many challenges. So over the period of time, you please share your slides. And my screen is not showing that you are connecting, unfortunately. So you have to work over that. Yes, it's
Are you able to
perfect.
see my slides?
Yes, it's perfect. Go
Okay,
ahead.
thank you. So today I'm going to talk a little bit about technology wise, and later part I'm discussing about how I'm using these techniques in my research. So I'm talking about the application of multi-omics technology in deciphering brain functions and diseases. So basically, when we say the multi-omics technology, it will deals through various high throughput technologies like one of the genomics, where we study a genome, transcriptomics, where we study the transcriptomic profile of a genome, then proteomics, which we deal with the proteome and associated post-translational modifications like phosphorylation, ubiquitination, and so on. And recently emerging field like metabolomics, which deals with the metabolome of a cell or a tissue, which is given a time in a global and unbiased manner, and that is referred as a multi-omics technology. And we are using these kind of multi-omics technology to integrate the data to get a more biological meaning of our data, because one or another technique cannot be give a full picture of the system, whatever happening at the time, disease condition or any biological process. So in our how we can use these techniques, we can use these techniques to decipher various molecular functions, like we can identify biomarkers for a disease condition, we decipher the signaling pathways, then we can use to identify the drug targets, and same time we can use them as specific purpose for a molecular diagnostics also for these techniques. Several techniques can be used, like molecular diagnostic. Already genomics is playing an important role in the diagnosis of the cancer, and uh, signaling path drug targets also can be deciphered by using these techniques. So at Yenepoya University, we have a dedicated research center. We comes under the Yenepoya Research Centre, and I belongs to Center for Systems Biology and Molecular Medicine, which is a sub-center of the Yenepoya Research Centre, where we have a state-of-the-art mass spectrometry lab, where we have two dedicated mass spectrometers, Orbiter Fusion Trigrid and QTRAP 6500. So we used to do here particularly like quantitative proteomics and multiple reaction monitoring, which is basically a kind of validation platform. It's just complementary to our ELISA, which use antibody-based systems. Then we are also doing mapping the brain proteome, because still we don't know much about the brain, which is a complex organ. So this project we are doing with the collaboration of NIMHANS Bangalore. Then molecular targets for traditional and Ayurvedic medicines, because it is one of the mandate of our center. So identify the targets of the molecular targets of the traditional Ayurvedic medicines, so that we can molecularize them and we can have a proof of concept to use these medicines, because Ayurveda lacking the point. Then functional proteomics. When I say the functional proteomics, means it is like the post-translational modifications, like phosphorylation, ubiquitination, acetylation, and other post-translational modifications. Then metabolomics and whole genome sequencing is one part, which sometimes we also used to do whenever it's required. So this slide represents the whole process of the biology. Like we have a gene, and mRNA, it will translate into the mRNA, it will convert into the protein, and protein leads to the final end type products, which is known as a metabolites, which is mainly nowadays think as a functional molecule of the cell. So gene and mRNA will be studied by the genomics. So we can study by mutations, epigenetic modifications, post-transcriptional modifications by the study of the genomics. Some proteomics refers to the study of the protein expression and sometimes the post-translational modifications, and study of the metabolites is referred as a metabolomics. So we usually, most of the cases, we do the integration of proteomics and metabolomics, and sometime whenever it is required, we will also integrate the genomics in our studies. So proteomic system study of protein complement, basically proteomics as a systematic study of a complement of the protein into the cell, where we used to study the protein ligand interactions. Then we can use these techniques to identify the protein complexes, protein families, their activity and structural supports, like they belongs to the kinases or they belongs to phosphatases, or the mainly post-translational modifications, like several post-translational modifications are there, the proteins, and so on, that can be deciphered by using the proteomics approach. So why the proteomics and post-translational modifications studies are necessity? Because we know that one gene can code several proteins, and that proteins may be due to the alternative splicing, and subsequently these proteins also has one protein can harbor multiple post-translational modifications. Like one protein can be a phosphorylated, another same time that protein can be a methylated also, but they had a different functions. So it is necessary to study the post-translational modifications also. So like what is the difference between, there is a difference between the high throughput proteomics approach and conventional proteomics studies, where we use antibody-based ELISA or antibody-based Western blotting system or immunofluorescence or immunohistochemistry, where we required, if you see that left panel, where when the fisherman is fishing a single fish, like one protein. So we know the information about the protein, we are doing the study, we are trying to see the study. But when I say comes to the proteomics approach, one person comes to a net, and whatever the fishes are there in that pond, he took out that fishes. So in a typical proteomic experiment, we will get all the information about the whatever the proteins available at that time of the system, so that we will get a huge amount of the proteins, and based on that we will do further see whatever the proteins which we are interested. So that is also comes under data analysis, but it will give a tremendous information about the protein component of the cell. So why we need proteomics basically? So proteomics basically we can use proteomics because we cannot deduce the protein alteration by the studying of DNA, because the DNA has a single there is no modifications of DNA or post-translational modifications which we cannot identify by the studying the DNA. Then RNA expression does not always reflect the protein levels because RNA also goes under so many post-transcriptional regulations, like it has a translational control also, it degrades, and turnover also affect the stability of the RNA. So every time we cannot get a complete information by studying the RNA also. Sometimes it is not suitable for RNA expression analysis because due to some tissue they had a more RNase contents, or maybe they are not practically good, feasible to study the RNA expression studies. And the proteins basically, it is a more stable moiety in the cell as compared to the RNA also. Then proteins are the physiological and pathological active key players, and it is easy to translate the results from the protein to the clinic. Maybe it is a diagnostic purpose, or maybe it is a therapeutic purpose. General goal is a better understanding of the genesis and progression of disease that can be studied very well by using the proteomic study. And clinical goals, early detection of the diseases, basically biomarkers. So in the scenario of India, it is still not feasible for go every time to the RNA-based biomarker or the DNA-based biomarker because it has technology-wise also not feasible, and economically also not feasible from the Indian point of view. Then identification of therapeutic targets and therapy monitoring. This is very important thing nowadays, because when we talk to the clinician, their first question is there whether I can able to monitor the therapy. They want to like some test which is reliable and less expensive in terms of the patient point of view, and give the monitoring of the therapy. Like if they are doing in terms of the cancer, if I'm coming, like if they are giving the chemotherapy, they can see the monitoring the chemotherapy, okay, it is working. They don't want to tell the patient every time go to the scanning. So yeah, this is the like where the proteomics can play an important role. So how we will do the proteomic experiments? So it is like a high throughput sophisticated experiment, and once the experiment is done in a properly, and if you get the experiment very well, so you can have a tremendous information, and that information after analysis give a clear picture of the system. So what the experiments will start from the very beginning, where we have a tissue or the cell lines and we extract the proteins. After that, we have two approaches to follow that. One is the gel-based fractionation system, and another one is a basic RPLC-based fractionation system. So where one sample we load on the SDS page, and follow up run the SDS page. We stain them, and after the staining we cut the bands and do the tryptic digestion, and then we run onto the fractionate again to the PLC, and we run on the mass spec. Another one, we say that it is an in solution digestion, where we use a trypsin or other cleaving enzyme in a tube, and followed by the basic RPLC fractionation, and the mass spec followed by the mass spec run, and then data analysis. We generally use two different techniques, because our aim to fractionate the proteins, so that we are our end target, the identification will increase, because both the techniques are having a different way of fractionation. This basic RPLC is a HPLC based fractionation, where we are going into the high pH, so that we will get the more proteins which is more abundant at the high pH. And SDS based, it will be basically separate the proteins on the charge to mass ratio. So both the techniques, when we integrate it, we will get more identification in the proteomic experiment. So this is like how the proteomics mass spec will detect the proteins. It will not detect any peptide, or it will not detect any protein. It will detect a mass to charge ratio basically. So when the one peptide will be there, it will be cleaved by the enzymes in the small parts, and in mass spectrometer we will provide a high energy in terms of high voltage. So high voltage dependent energy collision will be there, and it will again break down into the different parts, and this will be these different ions will be captured by the mass spec, and based on that ion and charge ratio, we will get a mass spec profile where you can see Y means Y and B will the ions, different ions are there. It depends on the cleavage of the peptide, at which point that is cleavage. Based on that, the Y ions and B ions will be generated, and this will give a clear spectra of the particular peptide, at which time basically which it is acquired into the mass spec. So this spectra basically gives this peptide will gives this kind of a spectra into the mass spec. Later, whatever the data we have again got this by the mass spec, we will run on a software, and we will get the list of the proteins along with their relative expression and other things. So this is in general chromatogram. While we will give 120 minutes run, so it will come. It will give a complex chromatogram. And this complex chromatogram, if you see one particular time point, like if I want to see that whatever happening at a 61.2 minutes, so this will be the basically 61.2 minutes. When I zoom it, it will be subsequently divided into the several different peaks. So every peak will give you the one ion. So an average global proteomic experiment by using LC-MS-MS approach. When we say the LC-MS-MS is a liquid chromatography mass spectrometry based tandem mass spectrometry, where we are going to detect the proteins at MS2 level. So we can identify whole proteome of the cell or tissue by fractionating our sample between 30 to 60 fractions, but that is a theoretical approach. But sometimes it will not feasible or not practical. So generally we will go till 12 fractions at least minimally, and each fraction analyzed by LC-MS-MS experiment. Then whole experiment will be surveyed like approximately 10 lakhs to 20 lakhs of the peptides, and we will able to identify total in between 2,000 to 10,000 proteins in a single experiment. Depends on the sample preparation and handling, and time for sample preparation overall it requires 3 to 4 days, and mass spec runs again it will require 2 to 3 days if it is run a 24/7, and it depends on the fraction numbers, and data analysis which is required approximately 1 to basically if everything will be fine in a 3 months time, we'll have ample information that can be further validated by other complementary methods. So we can do the relative quantitation also and absolute quantitation, because nowadays it is a more requirement that when you are using different conditions, like for example, if I am want to see the effect of a drug on a particular disease, then I had definitely I had three groups. One is a control group, one of my disease control, and another one is a drug treated group. So if I want to compare between these three groups, so that I want to include something which can give me the absolute quantitation or at least or relative quantitation. That can also be done by using the mass spectrometry based assays. And for that we have two different kind of approaches. One is a metabolic labeling approach, where we use SILAC labels, which is known as a stable isotope labeling with amino acids in cell culture. So basically this technique is specific to the cell cultures only, where during growing the cells we use heavy isotopes, which is known as a SILAC isotopes. So basically it will be like one amino acid which is labeled, and we will select the media which is a labeled amino acid media, and we feed the cells with the labeled amino acids. So for that 4 to 6 cycles of the growth, so the cells will be growing and they will incorporate with heavy, since it is present into the media, they will incorporate this heavy carbon and nitrogen, and mainly carbon and nitrogen into their building blocks, so that their whatever the proteins we will getting them after the lysis, they are labeled with particular SILAC. And since they are heavy isotope, so their mass will be different, and the mass spec will capture their mass. Then another one is the chemical labeling. Chemical labeling method. So chemical labeling method is a little bit straightforward, where we can use this kind of labels. Basically they are basically a tags basically. So it is like a chemical tag. Normal tags whatever we use our day basis things, like to write anything on things, like sometimes we keep into the book inside and book tags and other things. So these are basically chemical tags. So we will label them with the peptides. Now the chemical tags are different tags, like one is iTrack, another one is a TMT. Nowadays a TMT is a commonly used tags. And advantages of using the TMT or iTrack is that we can compare 10 different conditions at a time. So these tags basically had a difference between 1 Dalton, and for TMT 6 plaques means that comes under the 6 conditions, different conditions. So they having the 1 Dalton difference. But when you are going for the TMT 10 plex, they had a 1 Dalton and 0.6 Dalton difference. So that difference can be picked by the mass spec, and we will see that whatever the peptide will come which having the which tag, and based on that the mass spectrometry based software which is comes with the system can differentiate the peptides. Okay, this peptide comes from this tag, or we know that I have tagged my particular disease condition with what tag. I can segregate the proteins based on that, and based on that we can do the relative expression also. So quantitative proteomics can be used for identifying the mainly the identification of biomarkers and therapeutic targets basically, because when you are going to identify new biomarker, you cannot be a biased. Your approach should be an unbiased manner. Like if I want to see the biomarker for the early detection of the Parkinson or Alzheimer, or I want to differentiate between Alzheimer dementia and normal dementia. So I cannot be biased when that, okay, I want to see that only these 5 proteins, what happens to these 5 proteins in between 2 conditions. Then might be I am wrong, because might be these they are the very close associated disease, and might be whatever the proteins I am selecting, they are basically expressed in both the conditions. So in that particular scenario, we will go for unbiased approach, and we will take the serum sample, or maybe the cerebrospinal fluid, or maybe sometime it is a urine sample also. Between the different patients of different groups, we will follow in the mass spec, and whatever the data we got, we will just analyze it, and we can find out the whatever the proteome is there at the current time into the serum, into both the conditions, and based on that we can also found that differentially regulated proteins. Might be some proteins we can found which is exclusively present in the dementia, early dementia, but they are not present in the late dementia or maybe the Alzheimer dementia, then we can say that this is a perfect marker for differentiate in both the conditions. So that can be a very good tool to identify a new biomarkers. Sometimes then feasibility of the mass spectrometry for diagnosis, that is one of the major challenges nowadays, because still in India, mass spec is not meant for the diagnostic purpose. It is only for the research purpose. So if you identify the markers based on the mass spectrometry assay, then next challenge is that we have to convert them to the antibody-based assay, and sometimes the sensitivity will not enough, because mass spectrometry assays are more sensitive than antibody-based assay. So most of the things, it is a very challenging when we are working on the mass spectrometry-based biomarker discovery. So if you get 10 proteins as a biomarker or as a differentially expressed, I'm telling the differentially expressed proteins, finally we can conclude 1 or 2 proteins which can be used as a kit-based diagnostic marker because due to the sensitivity. So this is one of the challenge by using the mass spectrometry-based biomarker discovery. Apart from that, we can use a tissue proteomics also, secretome analysis, one kind of things which is very helpful. Like sometimes, especially in cancer, this kind of experiments are very important, where we can see every time we cannot go and see the patient tissue or kind of things, or maybe so we will see here when the cells, when the cancer cells will grow into the culture system, what they secrete into the surroundings. So we will take the media, spent media, and we will do proteomics analysis, and we can identify several proteins which is in a secretory nature. Then we just find out these proteins into the neighboring fluid where the cancer will be there into the body. So that is a secretome analysis that can be useful in mainly in the cancer research. Then proteomics from the biological fluids, several serum, CSF, urine, saliva, even bile salts, bile juice also can be the proteomics can be done. Phosphoproteomics, again, it is a post-translational modification. Glycoproteomics also the post-translational modification. Other post-translational modifications can also be done by using these approaches. So coming to the post-translational modifications, because my main interest is the identification of the post-translational modifications and the signaling pathways during the neurodegenerative disorders. So basically, the post-translational modifications will determine the cellular distribution of the proteins, because all we know is that once protein might be unphosphorylated and it present in the cytoplasm, but when it is phosphorylated, it transported to the nucleus where it can use as it can be work as a transcription factor, or it has several other functions. And that can be modulate the other functions of the cells also. Then activation and deactivation of signaling pathways. One protein or one protein upon post-translational modification can activate or deactivate other signaling pathways. Then mark proteins for the degradation, the ubiquitination pathways, which is very important into the neurological disorders, particularly Alzheimer's and Parkinson's disease. So till now we have several post-translational modifications which is known, and that can be detected by the mass spectrometry-based assays. One is common is the phosphorylation. We have phosphorylation on phosphotyrosine phosphorylation, phosphoserine threonine phosphorylation, then methylation, acetylation, ubiquitination, hydroxylation. This is a common post-translational modifications. Apart from that, we can use sumylation, nitrosylation. Other things can also be done by using these platforms. So we can use we can identify multiple post-translational modifications into the single samples by using a serial enrichment process, and we can these are the post-translational modifications and their corresponding difference into the mass. Basically, so phosphotyrosine and phosphoserine and threonine, basically they are combined, and they will give a difference between 79.966 Dalton. So it is easy based on the mass spectrometry based identification. We can differentiate between the unphosphorylated and phosphorylated, because 73 Dalton is quite good amount of mass to detect by the mass. So acetyl lysine basically 42 kilodalton, methylserine is a 14 Dalton. So these are the basically different molecular weight Daltons in molecular weights, which can be detected easily. Segregation can be done between unmodified versus modified amino acid. So now I'm going to the next part where how can the phosphoproteomic strategies to identify aberrant signaling pathways and potential therapeutic targets in neurological disorders. So phosphorylation is the commonest why I'm selecting the phosphorylation initially, that it is the commonest phosphotranscription modifications involved in the regulating the various cellular processes, like most of the processes, like if it's a 70 to 75% signaling pathways, it governed by anytime, any level by the phosphorylation, dephosphorylation events. Phosphorylation serves as a surrogate to determine activation status of the various proteins and also to monitor the kinase activities. Deregulation of kinase signaling pathways is associated with various diseases, including the neurological disorders, where we can find several kinases like CDK5, or you can say GS3 beta, or other CDK4, CDK6 are dysregulated, and it is very prominent in the cancers also, where several anti drugs which is currently available into the clinic, they are basically kinase inhibitors. Then mass spectrometry based quantitative phosphoproteomics allows us to investigate the aberrantly altered signaling pathways, and then we can identify the novel kinases which is altering, and we can go back and see the kinase library, and we can identify new molecules which can be used as a drug target or newer drugs for the various diseases or disorders. So in this way, it will help us. It will help a lot of us in designing and execution of the experiments. So this is again various enrichment strategies for the phosphorylation experiments, phosphoproteins, which basically we have several enrichment efficiencies. One is like we have a antibody affinity based enrichment techniques. We have a metal affinity based techniques, and then IMAC based affinity based techniques. Also, it depends on the your experimental choice and your outcome as we will select them. So this is about the phosphoproteomics. Now the third will be the metabolomics, where nowadays we are more focusing about the metabolomics, because it is the final readout. Initially we said that the proteins are the final readout. Now the metabolites are the final readouts, because the metabolites, they are very small molecules then produced by the cells, and they are basically each and every biological reaction is governed by the metabolites. Either they are serving as a cofactor, or they may be the end product, or maybe the starting molecules. But the meanwhile, they are very important molecules, and it is good to study them. A lot of challenges are also there. Because of that metabolite, they had a very structural diversity and chemical properties, and the thing is that till now we don't have a much good database where we have found significant amount of metabolites to prove our data. So several classes of the metabolites, which is very important metabolites. You have tyrosine, which is basically involved in the neurotransmitter dopamine biosynthesis. Then glucose, sphingomyelins, phosphocholine, they are basic cholesterol. They are important for the our membrane structure. Then other threonine and other, these are the amino acids, succinate and malic acids are also the important metabolites, which is involved anyway in the various biological functions. So basically these are the various classes of the metabolites. It is just representation of the various classes of the metabolites. And so we had a lot of challenges in the metabolomics also. So metabolomics basically they are basically comes in the very classes, which is very chemically diverse classes, and hard to develop a single technology wise. It is hard to develop a single, because the mass spec single chromatography method, it would enable us to monitor these diverse classes of the molecules. So as compared to the proteomics, since proteomics they were mostly the negative charge, and we will, by treating them with the SDS and other things, we will provide them a uniform charge. So it is easy as to do the fractionation. We can just go for the reverse phase liquid chromatography, and we will get a optimal results. It is not true for the metabolomics study, because most of the metabolites, they had a polar, non-polar, and neutral molecules. So based on that, we have to standardize every time. We have to standardize our chromatography method, and based on that, only we can identify a good amount of the metabolites into the given conditions. One thing is that another challenge is that for proteomics, we have a good databases like UniProt or NCBI database, which is very established database. Every time it will be updated a weekly basis, so that whatever that still we get a significant amount of the proteins which is identified into the system. Although in proteomics also we get several proteins which is non-identified, which is like the software will use them as a like proteins, like actin-like proteins, which has a similarity with actin, but it is still not discovered properly. But for metabolomics, we don't have that kind of databases. So whatever we identified, when I experiment, we identified at least 6,000 to 7,000 metabolites, but ultimately we end up with maybe 1,500 metabolites, because our database, only this much of database we have, which can map the metabolites. So since they are very different classes of the metabolites are there, generally people in the world, they use different kinds of technologies to fractionate them. One is like helix-based technique, and another basic RPLC-based. So helix-based basically use aminopropyl columns. So basically it will be helpful into the polar metabolites. And RPLC basically use the C18 columns, so basically it will be the hydrophilic metabolites. Basically, we can identify in using these columns. So basically, using these two columns, it is like we will increase our the number of metabolites, which is basically because number of metabolites, because we are at initial stage we are doing untargeted, unbiased manner. So whatever the maximum number we will get from the experiment, it is good for us. So the difference between these two strategies methods, like if you see that when you see the ESI positive mode, means the samples are run in the positive polarity of the mass spec, and minus means it is run in the negative polarity. Now, if you see that if this veni, so in the positive mode, the 28% metabolites, which is specifically I am identified into the helix column, which is not present into the RPLC, and 38% metabolites, which is specifically identified into the basic RPLC based fractionation. Same thing will be there in a negative mode also. So, so if I'll using a single method, if I'll go for RPLC based method, I may lose this 28% metabolites, and again I will lose this 43% also. So a significant amount of the metabolites I may lose if I'll go for a one method only. So this is one of the challenges in the metabolomic experiments. So this is all our workflow, how the metabolomics, typical metabolomics experiment will be done in an untargeted manner. So initially we extraction is based on various methods. But mainly we use methanol, acetonitrile, and H2O mixture for extraction. Then once the extracted will be done, we have reconstituted acetonitrile and water. Then LCM SMS based run, then software, bioinformatic analysis, and everything will be done. Ultimately, we'll get the metabolites. Then we will do the validation of these metabolites by multiple reaction monitoring or selected reaction monitoring. This is again a mass spectrometry based method where we know the metabolite, or we know the metabolite, or we may know the protein. So now we are looking, this portion will deal with the untargeted or unbiased metabolomics. Here we will know the metabolite, and we are just doing the validation of that metabolite. So, okay, I know that citrate is present, or maybe I know that dopamine is present into my sample. Now I just want to validate that dopamine is there. I just see the dopamine into my sample. So that is known as a multiple reaction monitoring, and that can be further extrapolated, and we can develop the representation will be done by like a box plot or maybe the diagrams. So another experiments which our ongoing projects is like we are interested to see the brain proteome of humans. So we collaborated with the NIMHANS, Bangalore. The difference, the idea behind that, we can see the differential proteomic profile of different subregions of the brain, and our group at CSBMM is involved in that, and we have got almost 32 different brain regions from healthy cadaver humans, and we have done almost done all the proteomic analysis, and we are doing the data analysis. So basic idea is that whatever the differences there into the brain, different reasons can be used as a biomarker or can be used as a disease progression marker, because these proteins, when altered, they can come into the circulation. So our aim is to that first we study the brain proteome, then subsequently we will want to looking these proteins into the serum or urine of the patients. So one example, this is I'm not involved in this study, but since I am discussing about the biomarker and mass spectrometry based assays to discover the biomarker, so I'll just represent one or two examples where our group initially, before joining to me, they were involved into the identification of the markers of acute ischemic stroke and another type of stroke, which is a because the window period between the stroke is maybe that 1 hour or 2 hours based on the clinician assessment, and that window period is very less to diagnose, and the diagnosis of the stroke is depends on and diagnosis the treatment of the stroke is depend on the drug. So selection of the drug is depend on the which type of stroke is there, whether it's ischemic or it is a vascular. So if it is a stroke, so we have, our group has initially done proteomic analysis of the serum samples of this different kind of strokes, and they have identified 2 proteins which is exclusively present in the ischemic stroke. That one is there, Wellbrand factor, and another one is ADMTS13 protein. So they have identified this protein which is highly expressed into the stroke conditions and which is very less present into the control samples. And later these findings they have validated on the other set of samples by using the complementary method with ELISA, and the same thing which is we have observed by the ELISA, and this is basically turned to be positive in ELISA also. Now this group are doing a larger scale of the clinical trial for this protein as a biomarker for early detection of the ischemic stroke. Same story will be the ADAMTS13, which is basically a disintegrating and metalloproteinase with a thrombospodinin type 1 motif member 13 protein. Its name is quite large. And the same thing will be there for this protein also. However, this protein is expressed very less into the ischemic stroke. So these 2 proteins will now we have taken up for larger cohort, because initially we have done 35 or 50% cases only. So this is one of the example where we can use mass spectrometry based proteomics to identify the new biomarkers. So this is till now I have discussed how these especially the proteomics of phosphoproteomics and metabolomics can be used or whatever their approaches. Now how I use this thing into my studies, like so one of my work which is basically depends on the identification of molecular targets or signaling mechanism associated with the traditional Ayurvedic medicine. We use a common Ayurvedic medicine which is known as a Yashti Madhu. Maybe the north people easily know it is like Glycyrrhiza glabra, and it is basically used for the Ayurveda some other purpose, like it is used for the sweetener, or it is like explorant for the throat infections and other things. And if I'm not wrong, in the north at least the people will give it to the small childs for the proper voice. So this is known as a Glycyrrhiza glabra. It is an botanical name, and we sought to see that its neuroprotective functions in Parkinson's disease model, which is basically rotenone-induced Parkinson's disease model. So we use proteomics, phosphoproteomics, and metabolomics approach to identify overall how this brings to the neuroprotection into the rotenone-induced Parkinson model. So this is all about the experimental process, experimental workflow. Like we use IMR32 cells. Since these cells are the neuroblastoma cell lines, so before we start the experiment, we have differentiated them by using retinoic acid. So upon differentiation, they turn to the neurons, and then we treat them with rotenone to generate the PD model. After that, we treat the cells with the ST-Madhu, and we will perform different experiments. Like one, we do the targeted and untargeted metabolomics, then proteomics, and followed by the phosphoproteomics. So before this is the overall experiments. Once I got this drug from one of the collaborator, which is associated with our Ayurvedic hospital. So I just had a curiosity whether like if I'll go directly, because he came to me that it is an Ayurvedic text, it is written like he has some memory enhancer properties and other things. But if I'll go directly by using this drug, and at the end of these sophisticated experiments, if the end I didn't get anything, then it is very setback for me. So I started that untargeted metabolomics of the ST-Madhu root extract, which is basically root powder. So we took the root powder, and we have done the metabolomic analysis, and we identified several metabolites, like some around 1500 plus metabolites we have identified, and they belongs to various classes, which is the typical classes of the metabolites like di and triterpenoids, alkenoids, flavonoids, and so on. And then we will do some bioinformatic analysis. We have seen some, like we have identified the metabolites, then that metabolites we have mapped with the human proteins, whether they have some interaction with the human proteins. Then based on that, the metabolites which shows the interaction with human proteins, we have segregated these human proteins, and we identified certain pathway enrichments we have done. And we have found that these metabolites, which is coming from the ST-Madhu root extract, has interaction with several key pathways, signaling pathways, which is basically involved either in the neuronal neurodegenerative disorders or mainly in the neuronal functions, differentiation like MEK-ERK pathway, then P38 MAPK pathway, PI3K-AKT pathway, cell cycle related pathways, GPCR signaling pathways. Sometimes we also seen that whether these metabolites can regulate the neurotransmitter receptors. They had an interaction with neurotransmitter receptors. So we have identified almost all type of neurotransmitter receptors like dopamine, serotonin, bradykinin, P2Y receptors, then adrenoreceptors. They have showing the interaction. But my point is basically I'm interested in studying the Parkinson disease model. So if I found the dopamine receptors are there and these metabolites are interacting with dopamine receptor, we go through further and we have done the proteomics, phosphoproteomics, and metabolomics to see the whether these findings, which is basically a bioinformatic based approach, whether it is correct or not. So we use multi-omics approach to unveil the neuroprotective functions of the ST-Madhu. So technically I use quantitative proteomics, phosphoproteomics, and metabolomics approach behind this neuroprotection. So initially we have done a proteomic approach. So proteomics approach we have done in 2 biological replicates, and we use since we have 4 groups, we have used 2 different sets of labeling experiment. We use 4-plex TMT label, and since we have done the mass spectral analysis, so we have identified total 30,554 proteins across the this number is basically a pooled number across the all 4 groups, and this leads us to the 4,864 proteins. Since we are interested into the since we have run 2 biological replicates, so I want to give more confidence in my data. So I use I went common proteins across both the biological replicate will give the 3,762 proteins, and ultimately I filter based on the PCMs and further filtering will be done. I conclude the 3,720 proteins, which is I identified across the 4 sections, and later we will see them, and out of these 4,000 proteins, 4,000 plus proteins, only we have identified some around 160 proteins, which is 421 proteins, which is differentially regulated upon rotenone treatment, and for one data also 144 is significantly altered. and 144 significantly altered proteins, which is basically a rotenone treated conditions. Out of the 84 proteins, only we have identified which is significantly altered upon the rotenone Yashti-Madhu treatment. Means these 84 proteins are basically involved in the neuroprotective functions of the Yashti-Madhu. Then we map these 84 proteins, and we identified these proteins belongs to the several key biological process which is involved in the Parkinson disease. Like they belongs to the mitochondrial proteins, endoplasmic reticulum, then DNA repair and replication, cell cycle, oxidophosphorylation, autophagy, and TCA cycle. So, and E3 ligase is one of the families there, which is involved in the ubiquitination. So these are the proteins which is altered by the rotenone, and these are the proteins which is restored by the Yashti-Madhu. So most of the protein which is altered by the rotenone, these are same belongs to the same family, autophagy and mitochondrial dysfunction, autophagy, and cell cycle related phenomena. So then we will go for the, since one of the proteomics, and then I will just want to see that what is happening to the metabolome level. So the same experiment we have repeated with the metabolomics experiments, and we have identified several metabolites. Like total we identified 2,403 metabolite from positive mode and 2,934 in negative mode. Out of that we have only able to map around 1,100 metabolites. Out of this around 5,000 plus metabolites, we have identified only the 1,100 metabolites which can be mapped to the non-metabolites. Remaining will be, we don't know. We know that these are the metabolites, but we don't know which kind of metabolites we have because we don't have the database to map. Then our picture will next will identify that out of these 1,200 plus metabolites, only we have identified some 200 plus metabolites which will be the altered upon the rotenone treatment, and out of this only the 88 metabolites which we identified which is restored by the Yashti-Madhu. So again these metabolites belongs to the several pathways from the human cells. Like one of the English pathway is the citric acid cycle pathway, another is the tyrosine metabolism pathway, and mitochondrial electron transport chain pathway. So based on this pathway, we have selected for further analysis, which we have done by the MRM method, multiple action monitoring method. This diagram will show that since we have done already the proteomic analysis, and now we will want to compare our metabolic analysis. So whatever the metabolites we have identified which is differentially regulated upon rotenone treatment, and when they differentially regulated restored by the Yashti-Madhu treatment, we will going back to our proteomic data, and we will assign the metabolites to the proteins, which proteins they are involved, they are regulating based on the bioinformatic approaches. And we identified that several proteins which is available into our proteomic data set, like citric acid cycle pathway, ubiquitination mediated pathway, then pyruvate metabolism, pyrimidine, thiamine, they are altered upon the rotenone treatment, and most of them, specifically the citric acid cycle, then glutathione metabolism, are restored upon the Yashti-Madhu. So this will give the our analysis like a positive feedback for my analysis that whatever I have done, it is correct. So later we will construct a metabolite protein network map where we can see we have put all the metabolites which is we identified and the metabolites which proteins corresponding to these metabolites. So like if you see that here, this yellow triangle shows the metabolites regulated by rotenone only, and red square the proteins regulated by rotenone only. Then green purple triangle will be metabolite restored by Yashti-Madhu, and blue will be the protein restored by the Yashti-Madhu. To see if is this metabolite lanthionine, basically it is regulated by the rotenone. However, this coenzyme A is also regulated by the rotenone, but this coenzyme A will interact with another metabolite. This is a pertussin, which is basically regulated by the Yashti-Madhu, and this metabolite ultimately regulated by a protein which is regulated by the Yashti-Madhu, which is an OAT protein, and another set of that protein is HMGBT. Like in this way, we have constructed whole map where how the proteins and metabolites can give the crosstalk basically. So in this, based on the crosstalk, which is I am not included a data due to the times concern, we have come out to know that that the dopamine biosynthetic pathway, which also altered in rotenone induced abbreviations in the Parkinson disease model, and which is basically revert back to the normal levels by Yashti-Madhu. So coming to the phosphoproteomics part, the same approach we use for the phosphoproteomic experimental approach will be the same. However, since previous both the experiments has done at the 48 hours of the time points. However, that since phosphoproteomics, we wish to see the early signaling mechanism, at what point that signaling will established. We use early time points, which is 2 minutes, 5 minutes, 15 minutes, and 30 minutes of the post treatment. And we have done it in the 3 biological replicates. We use 2 methods of enrichment, which is like TiO2 based enrichment and nickel iron NTA based enrichment. So to get the more proteins, phosphoproteins into our study. Then this is the whole story where we identified the different time point based different proteins. We have identified phosphoproteins we identified, and they are differentially regulated. So based on that, if you can see, the most of the proteins which is differentially regulated, they falls under 2 minutes and 15 minutes time points. So based on that, we can conclude that whatever the signaling mechanism is there, they mainly established between the initial 15 minutes of the time. After that, just the protein level will change. So based on that, this is basically the proteins which is altered upon Yeshimadu, and this is dysregulated. This is upregulated upon this is downregulated of the proteins, which is upregulated by the rotenone and downregulated by the Yeshimadu. And this panel shows the protein which is downregulated by the rotenone and upregulated by the Yeshimadu. Based on these, we have again make a kinome map. We try to put these proteins, which is we identified exactly, which is involved in the neuroprotective functions of the Yeshimadu, at where they will sit into the kinome map of humans. So we have just put whatever the proteins we have got, we have kept them into the kinome map, and we can see that they are basically involved in the several well-known kinase family, like serine-threonine kinase family, then protein kinase AGC kinase family, and then CDC-like kinases and other things. So these are the basically this just we can do to see that whether we are going into the correct direction, whether we have like GSK3 beta, they are come together like that. So based on that, we have just this map will show the kinase substrate interaction map, where whatever the protein we have got, we have tried to, if it is not a kinase, it is just phosphorylated protein or it is a receptor. So we try to identify its upstream or downstream kinase target. So and then once we got that information, we have tried to make a map, and this map shows like purple color will show, or light bluish color will show the kinases that not regulated by Yeshimadu, and red color will show that kinases which regulated by the Yeshimadu, and green box will show the kinase targets regulated by the Yeshimadu. So like see, these are the kinase which is not regulated by the Yeshimadu, but like GSK3A, which is basically regulated by the Yeshimadu, and its immediate downstream target will be the LMNA protein. Same thing like a TGF, BR2 and PRKCT, these are the regulated by the Yeshimadu, but their downstream targets are there. These protein, the downstream target also there. So in this way, we have make a map. But my again next target is to integrate my proteomics, metabolomics, and phosphoproteomics data into the single experiment. So we took all the 3 data sets, and we will do all the bioinformatic analysis. We have a proteomic results, we have a metabolomics result, which is translated to the proteins, and then we have a phosphoproteomic results. So we have put all together, and we have made a early signaling network map associated with the neuroprotective functions of the Yeshimadu, where you can see that we have put the metabolites also, like dopamine, which is regulated by regulated D2DR3 receptor, and which will subsequently activate one is a phosphatase which can be activated by it, and subsequently it will be regulated the other processes into the cells, like ribosomal biosynthesis, cell cycle, and autophagy, and cap dependent translation and protein synthesis, etc. So these are the various biological process which is regulated by the Yeshimadu. Some findings which is not involved in this map, which is since I thought that time is not permit me, so that I didn't put it. But the one basically which is we had a key findings which is we have submitted for the publication, and it's under review. So that there is one more reason which is I have not put it here. We have identified since mitochondrial dysfunction, we have identified, if you remember in our proteomics data, so we have tested the TCA cycle for the metabolomics, because TCA cycles is also enriched in the metabolomic results. And based on that, we have identified that TCA cycle will leads to the regulation of mTOR-C2 pathway, and mTOR-C2 pathway mediated autophagy. So these are the all kind of things which we have concluded here. Like based on that, I had initial information that one information which comes through a Ayurvedic practitioner, that Yashtimadu can be a neuroprotective in maybe in a Parkinson disease model or in the Alzheimer disease. And he want to test the things, and he approached us, and we just took that because my interest is also there. So I just go through that these three techniques, and based on these three, I can conclude that yeah, Yashtimadu brings neuroprotection. It has a neuroprotective ability in Parkinson's disease cellular models, and via it regulate the cell cycle. It regulates via apoptosis, TCA cycle, energy stress, autophagy, and it also helps in the restoration of the dopamine levels, which is basically replenished upon rotenone treatment, which is a hallmark of the Parkinson's disease, because in Parkinson's disease, the dopaminergic neurons are dying, and we don't have the tyrosine hydroxylase enzyme, so it will convert tyrosine to dopamine. But that story is still not complete, because I don't have the reason why it is happening. Unless until the reason will not be there, I will not confident about that results. But yeah, still I can say that it will prevent the neuronal apoptosis via modulating the cell cycle regulation, TCA cycle, and energy stress, autophagy. So like so many process in this will be the regular process of the phenomena, like autophagy, and then TCA cycle, cell cycle, are regular process which will involve in the day-to-day functions of the neurons. So therefore, it is like we are proposing that there is a fine balance should be there in these processes. Once the balance will be disturbed, we see more apoptosis and autophagy, and that will ultimately lead to the mitochondrial dysfunction and apoptosis. So Yashtimadu brings that fine balance again, and in this way it will protect neurons for the dying process, which is due to the Parkinson's disease. So based on that, I am just conclude. I will thanks to my fellow team, which is led by the Dr. T.S. Keshav Prasad, who is a deputy director here, and the students who has worked directly or indirectly on this work, like Gayatri K. She most of the work which is I have presented here has done by her. Muhammad Altaf will be the he used to manage our mass spectrometry facility. Then Sandeep and Santosh, both are our bioinformaticians. They used to help us in the pathway enrichment analysis and other analysis. And Rex and Ankur will be working with me, and they help me in the cell culture and related experiments. Most of the work has been done by the funding with the internal funding, which is provided by the Yenepoya deemed to be university. And yeah, we have special thanks to the Karnataka Biotechnology and Information Technology Services for infrastructure support to the Center for Systems Biology and Molecular Medicine. Now, I'm happy to take the questions.
So, thank you, Dr. Modi. You have given a wonderful talk, and the present scenario of the multipic approach is viable. And it is always good to understand our biological molecules using different approach. There was a time when we understand our molecule with use of only 4 nucleotides. And new approaches are coming to understand these molecules, their functional diversity. But you said already all these technologies have limitations and they have pros and cons. But every problem has silver shining, and it opens the new window of research. So this talk is open for question. Now, yes, from the delegates, I would request you to place 1 or 2 questions before Dr. Modi. Otherwise, I will grab the chance.
Madam, should I ask one question?
Yes, sure.
Dr. Prashant, really it's great to see you giving a new insight to the traditional medicines. My question is, suppose if I want to get some metabolomics works done with my sample, so what is the prerequisite? Does it has to be a tissue sample, or should I have to send a tissue extract?
Anything is fine. It depends. It depends on your resources. If you have resources, it's better you can send tissue samples, but when you say the tissue samples, it should be accompanied with a dry ice and other things. But if you extract the metabolites, then it is easy to send. Maybe it is in a 4-degree also, or maybe sometimes if it is a extracted metabolites and dried samples, then might be you can send directly by the normal post also, because it is a dried sample. So it is depends on that. It is not the problem. Whatever the things should be taken into consideration, like that extraction method actually. Samples can be transported in any, even sometimes we say that the protein samples also sent by normal courier also sometimes, because we prefer to lyse it in SDS. So you know that protein, nothing will happen in SDS. Same thing like in the metabolite samples also. If had a facility of dry ice, then we prefer to send that tissue, or maybe the tissue mess mostly. If it is not there, then just follow the protocol, and maybe like first you quench the samples with liquid nitrogen, then extraction. And the extracted sample can be transported at even room temperature also, once they can dry, because they are an organic solvent and it is easy to dry. And after dried, extracted metabolites can be sent by the 4-degree or maybe in a room temperature also.
Another major cause of concern for the researchers is the cost involved. What can be the approximate cost per sample? Suppose if I want a sample to be, suppose if I want to perform metabolomics, you want to use the metabolics facility of your institute. So what will the approximate cost per sample?
See, that is again, I cannot answer that thing into the public forum actually. But yeah, I can say that whenever we have several collaborations, and the people who comes never disappointed. Okay.
But there will be some charges.
Yeah,
suppose if I want to get it done anywhere in India, so what will the approximate cost?
See, the approximate cost again, it approximate cost will depends on your experimental design and how want to you run. Like one thing, if you see the proteomics experiment or metabolomics, I just told that fractions. So number of fractions will increase, then cost will increase. So in that scenario, we used to go for like sometimes we say that 6 fractions is enough. If budget is less, then we can say 6 fractions, because whatever the information will got into the 6 fraction, we can little bit maybe 10% more aid into the when you go for 12%, 12 fractions. Same thing the metabolites also, metabolomics also. It depends on again how you want your data to be confident. Like sometimes people will come like, no, my data should be very confident, and they will prefer that they will run the sample into the 6 biological replicates and technical triplicates. Then cost will increase, but at the same time, the complexity of the experiment will also increase. If you have a 6 biological, we generally runs on a 3 biological replicates and 3 technical replicates in a positive mode and a negative mode. So that is enough. But sometimes the people say that no, my budget is not there. Then we will go for 2 biological replicates and 3 technical replicates also. That is also again feasible according to the system. Ultimately, your data should be the confidence.
Okay, thank you.
Dr. Modi, may I add a few things?
Yeah.
Yes. So one thing is that always the extraction of the metabolites is better for the analysis. And second thing is that if we do not go for the multiple replication, then issues come, because the availability of the data, comparison of the data is another issue for the metabolomic study or any kind of the study which is based on the online platform. That I understand. But anyway, it is very good to figure out the solution.
Thank you.
So this is the time to cap this session, and I am very much thankful to Dr. Modi.
Can I have a question with Dr. Prashant Modi?
Sir, yes.
Namaskar, Modi.
Namaskar, how are you?
Not bad.
Long time back I am seeing you.
Yeah. So actually I have a question regarding metabolite measurement, and not a question, basically suggestion. So say we have cancer cells growing in culture and we want to measure metabolites, say TCA cycle intermediates.
Yes.
What would you prefer, cell lysate or media, the media from which the media collected from cells? Because I've seen people using both, and also do you find if you do both, would you see similar trend or opposite trend?
No, if you do both, then you will definitely get a different trend. See, one thing,
Okay.
the people who's using both the things like TCA cycle and in cells and in a spent media also, they're they may be interested into see that TCA cycle metabolites are excluded from the cells. Like it maybe comes to the surrounding tissue fluid, and they might be want to develop it as a biomarker. That is the one of the point which I understand, because TCA cycle operates inside the cells, and we used to do the TCA because TCA cycle is a very common altered metabolite set. Okay.
Yeah.
And I have also done, which is like due to the time restrictions, I am not put it here because I am focused my talk in a general technology based talk, not my work based talk. So we also done the TCA cycle, and during this condition, and we also probe all the TCA cycle metabolites by using global and targeted metabolomics. And what I found, most of my TCA cycle metabolites, like with their intensity wise or their abundance wise, they are more abundant into the tissue lysate, cell lysate, and they are very less into the spent media. But yeah, if somebody will working on the cancer cells, that might be possibility hai कि they will be secreted from the cells to the spent media. So it is better if they want to see, they want to confirm that it's fine. But
so you are suggesting it's preferred to do cell lysate, right? Especially if
yeah,
I want
in my
to measure
opinion,
metabolites in the cells, right?
Cell. It is there in the cells mainly.
Okay.
Because it is very less chances that they comes from mitochondria to the cytoplasm, and then again it comes to the cells to outside the secretome.
Okay. Okay. Because I mean, we have a collaborator, and we were having this conversation last week only, and they were not sure what they are suggesting. Like they were not sure what to do.
Yeah, if they are not sure,
I have very limited expertise.
Yeah, if they are not sure, then initially they just go for the cell lysate. Might be after cell lysate, if they got something good amount like differentially regulated metabolites, different conditions, like maybe whole TCA cycle or maybe 1 or 2 metabolites from the TCA cycle, then might be it is easy to see that particular dysregulated metabolites into the spent media also.
Okay. Okay. So first pass should be cell lysate.
Yeah, in my opinion.
Okay. Thank you, Via.
Thank you. Thank you. Apart from the academic session, it was alumni session of Varanasi, BHU University. Anyway, okay, so it was a nice talk, and we would like to see some of the time. And now I am requesting to report here Dr. Payal Mohobiya, and it is now over to Dr. Mohobiya.
Thank you, ma'am. Good afternoon, everyone. I am a reporter for this session of today's international webinar on neurobiology of behavior, the challenge and the promise for translational research. In today's session, we heard 3 talks. The first speaker in this session was Dr. Meena Kumari. She presented her work on multifunctional proteins and their impact in neural physiology and addiction. She described the multifunctional proteins, specifically protein kinase C. She discussed that this multifunctional protein is a product of a single gene. She also described the activity of GPI and how this promotes the survivability. She told the range of alcohol limits, and that was 0.03% for Indian population and 0.8% for American. Late, she emphasized over the glutamate receptor on MMDA receptors. The information also provided us regarding G2 alpha and G2 beta and other proteins are very informative. The next speaker of this session was Dr. Deobrat Dixit. He gave a lecture on Epitranscriptomics, a new RNA code for the regulation of gene expression. Dr. Dixit discussed at length about new involving research area of RNA modification and profiling of glioblastoma cell and neural stem cells, which was at the same time initiated by his research group. He told us about the role of YTSDF1 and YTSDF3 protein that not affects the GSC growth, but YTSDF2 is essential for tumor growth, and also told us about IGFBP3 protein. And the last speaker of this session was Dr. Prashant Kumar Modi. He gave the lecture on application of multi-omic technology in deciphering brain function and diseases. He told about us omic technology aim to measure the total composition of a specific biochemical group, genome for DNA, transcriptomic for RNAs, proteomic for proteins, and metabolites for small hydrophilic compound. Multi-omic technique currently holds a promise to fill gap in the understanding of human health and disease. He also described the significance of phosphoproteomics for the identification of therapeutic target. We are very thankful to all the speakers for your talk. Thank you, ma'am.
Thank you, Dr. Payal Mahodaya. So now this is the time to thank you. I am grateful to organizing secretaries, Dr. Rajkumar Query, Dr. Divya Rawat, and convener Professor G.D. Ahi, head of the department Dr. Professor Subodh Sen, and senior faculty member Professor Varsha Sharma, who has given me opportunity to chair this session and become the participation of this important academic session. I am very thankful to all of the people, those are directly and indirectly associated to this webinar. Thank you so much. And now I am leaving the meeting. Thank you.
Thank you, ma'am. Now it is the time for lunch break. Please come back soon after 1 hour at 1:30 for session 5. Thank you very much. Please come back at 1:30 sharp.